Okay, welcome everybody joining us in the Zoom video conference and on YouTube. I think we're just hitting 1,000 viewers and numbers still rapidly going up, but we have a full program, so we want to get started now. Maybe at the beginning, a note of apology. This is an experiment. We've never done anything like this before. We've organized many conferences, but never such a big, purely online conference. And most likely not everything will work. So if you have trouble following, try to fix it or send an email to the contact email that's on our workshop webpage, or use the Google form that we use to submit questions, to send us any feedback. If there's something we can address during the workshop, we will try our best to do so. So based on the feedback we got, we have viewers from all over the world and also from a relatively broad background. So we would like to encourage the speakers, at least all of those that are already online, to try and make their talks accessible to a broad audience, because what we've learned is that this is a topic that's not just interesting to scientists, but it affects everybody on this planet. So I want to start with a little bit of introduction, but it's just going to be 5 minutes about ELLIS, about machine learning and AI, and why this is applicable, to make it plausible, why this is applicable also to fields like coronavirus research. And I want to start with this picture about the first industrial revolution, which was triggered by the steam engine and by water power, as we all know. And then the second industrial revolution was driven by electrification. So in a way, both of them were about how to generate and convert forms of energy. Now, the current change that we are experiencing has been called the digital revolution, but it really already started in the mid-20th century under the name of cybernetics. And in this revolution, energy was replaced by information. Now, like energy, information can be processed by people, but to do it at a large scale, we need computers. And like energy, information is probably a conserved quantity, so we cannot create it from thin air. Now, the first two industrial revolutions had major consequences. We all know that, and the ongoing one may be similar. And in fact, it's our information processing abilities, not our physical strength, that make us human, and that are the basis of human dominance on this planet. Now, like the industrial revolutions, we seem to have two phases. The first one was driven by computers and programming languages. The second one now unlocks a new information source by harvesting information from unstructured real-world data. And the enabling technology, and this technology will be applied also in corona research, as you'll see in the next hours. The enabling technology for this is machine learning, which by now is often simply referred to as artificial intelligence. Now, the ELLIS Network, which hosts this workshop, started 2 years ago as a grassroots movement driven by some of the leading European AI scientists. And what people had in common was that a certain concern that Europe will fall behind unless we act now. And we think we can still safeguard and develop European technological sovereignty in this field. And it's not just about money. It's about coming up with the right mechanisms to build excellence, which attracts and enables the best young talent. And it's about creating ecosystems where basic research, applications, and economical development, such as startups, work in synergy. And it's about how to make each ecosystem stronger by connecting them across Europe. Oops. Now, to realize this vision, we implemented a set of measures. So the first one are the ELLIS programs. These are modeled after the Canadian CFAR programs. They bring together a group of 10 or 20 of the top scientists in a field and work together. They work together, they meet several times a year on workshops to drive the development in one area, and I'll get back to that in a minute. Second component is a PhD and postdoc program. So we are in the process of creating a truly pan-European PhD program and postdoc program where people get to work together with several advisors of their choice, which we've learned is highly attractive to the top students. And we're very happy that both of these, the ELLIS programs and the PhD and postdoc program, will be supported by the ELLIS proposal, which was a proposal that we submitted together with and coordinated by our partners in Finland to the European Union under the Horizon 2020 program. So this will be funded with 12 million, and we'll use this to support these two measures. And the third one is that we're in the process of establishing a set of ELLIS units. So these are little ELLIS research labs within existing research institutions. And we made a public call for these labs last year, and we got an overwhelming response. A huge number of outstanding places wanted to start an ELLIS unit, bringing to the table their own funding. So we have several hundred million euros that are now being invested in ELLIS units. And we were planning to have a big celebration, a big event today on the 1st of April at the Royal Society of London. We had been planning this for months, organizing all sorts of things. Now, we all know that the world has changed since then. Obviously, we're not meeting in London today. And we're also not meeting to celebrate our ELLIS units. We hopefully can do this at some point in the future. But we are experiencing that the world has changed in our daily lives, but also in our work, and it affects all of us, or I think certainly many of us within ELLIS, also in the choice of research directions. So if I just speak from my own lab, I've learned that there's a huge creative energy and a huge motivation among people to contribute towards tackling and managing this disease. And for instance, in my lab, people have started about causality analysis of what are the drivers of different mortality rates, how different things affect each other. We've started thinking about how to use tracking data in a private way, etc. So there's a whole range of different applications, and things are happening very fast now, maybe not always with the same scrutiny that we are used to in science, but there's a big sense of urgency, and we felt it's now maybe a good time to convene this workshop because a lot of people know that stuff is going on, but they don't know exactly what's going on. And this workshop should facilitate presentation of what's the current state of the art in different directions. Where can we make a contribution? What kind of technologies are necessary? So that people in the audience can also see a match where they can bring their own expertise to the table. Now, coming back to this program, so we launched this set of programs across a variety of fields, some of them in basic research, methods development and theory, but some of them in application areas, such as one that's in the climate sciences. And we also launched one in the field of health, machine learning applications in health. And with the recent development, put this program at the center of our activities, if you want. And a lot of people in that area of health are actually actively working in that area, but also in other areas of ELLIS, but especially in this area are actively working to manage and contain this disease. And with this, I would like to hand over to Olli Stegle, who is one of the directors of the ELLIS Health Program, who will tell us a bit more about this program and about today's workshop. Thank you very much.
Yeah, I would really like to take up on where Bernard finished it with ELLIS Health, and also would like to welcome you all on behalf of the ELLIS Health Program, which is coordinated by Gunnar Vetsch, Michele Fanischer, and myself. And we have also a very, very diverse group of fellows, and many of them are also here on the call. So I think that will also be an interaction of shaping ideas and finding out what we're all doing towards this crisis. And some presentations will also come from our end, but most of those will from the community of machine learning and outside, and also domain experts. So just a few lines here about what ELLIS Health seeks to do. We obviously I'm just trying. Apologies, there's some audio hiccup here. I hope you can hear me now again. Apologies. And we obviously want to drive innovations in different areas and create a feedback loop between machine learning innovations and health sciences and human health in particularly, but also foster training and education of the next generation of scientists. And many of us are actually very close to the technology and driving technological innovations in different fields, from imaging to omics to genetic variation. And what really brings these applications to bear in the context of human health is interdisciplinary bridges. And this is something we really put in the center to really create such a bridge between AI and machine learning research and human health, and really the challenges that are happening in this field, in this domain, to really have an impact and to get the domain experts on board and have a sort of a feedback cycle backwards and forwards to create these bridges and make a difference. And actually, this workshop here today is one such bridge. We really hope that this helps also to create bridges between people who are interested in AI methodology with a health focus or not, and want to find out about the new challenges and questions that one could engage in the future or already now. And to motivate a little bit, I have here one slide that tries to make the point that there is an increasing number of different challenges. We all know the exponential numbers of cases going up across Europe here, in this case showing, and that will motivate a range of different machine learning and AI challenges. And some of them we can already tackle now. And we hear today about work going on in predicting outbreaks and tracking individuals. There's an enormous burden on the healthcare system, so healthcare management will be one area you'll hear about, but also the first step towards drug development, vaccine development, where machine learning methodology has traditionally an important impact, and of course also in this crisis. And this list of topics will continue as we go along in this crisis. It's very hard to predict how long it will last, but it's very clear that soon there will be additional data available, enabling more epidemiological research, host genetics of COVID and other elements. So I think the number of questions will emerge. And the main point of the slides is we are only at the beginning of a journey, and hopefully there will be also more opportunities to really tackle some of these questions using AI methodology. With this, I would like to point out that this workshop is a continuous activity. It's the start of several efforts that are ongoing. This has been really put together, as Bernard mentioned, as an experiment on a very, very fast time scale. So please bear with us. We want to make the abstracts and videos of the presentations available. We also want to click point resources such that others can engage later on, such as Slack channels, mailing lists, ongoing activities. And if you're aware of any activities we have missed, please do let us know. And you can also use the feedback forms we will make available to drive these sort of efforts and really make this webpage a vivid and active platform. And with this, I just want to finish here by providing you an outline of the program. We're very excited that so many excellent speakers signed up on such a short notice. This is really less than a week. The program is the whole development time beginning to end. We have some exciting work on outbreak prediction, viral host genome sequencing, drug development, healthcare management. And in the end, we also want to come back to this question about resources, challenges, and invite you all to engage and also have a very active discussion. So I just want to reiterate, this is a very broad conference and we have many people on the line. So to all speakers, please try to be concise, stay within your timing. We want to keep the question slot, which is not on here, which is substantial to get really discussion going, and obviously make sure everybody's understood. There's a Twitter handle here at the bottom if you want to engage. And there is a very small, tiny URL where again, you can submit your questions, which we will pick up on after every session in the question answer session. With this, I want to finish here the introduction and hand over to Bernard again, who will chair the first session.
Yes, so I'll try to make things very short to leave the maximum amount of time for the speakers. So our first talk will be given by Nuria Oliver. She's actually one of the people driving ELLIS, member of the ELLIS board, and also a fellow of one of our ELLIS, or active in one of our ELLIS programs. But in recent weeks, all her energy has been taken out by being a special, I don't know what's the title, special commissioner of the Valencian government to handle the COVID outbreak in Spain and working on a range of projects in that area. So I won't say much about her background. She has a PhD from MIT. She has been engaging in various ways in machine learning, also for charities and public activities. And I'll give it to her now. Thank you. You're muted.
Now it's like there was some kind of control that I couldn't unmute by myself. Okay, now I can do it. Well, thank you so much, and thank you everyone for being here. I'll try to go pretty fast mainly in my presentation. I'm just going to share with you how we are working in the Valencian region in Spain to use data and the analysis of the data to help make better decisions and help fight better the COVID-19 crisis. So as Bernhard said, my life before March 3rd was a very happy life, very involved with ELLIS, and particularly with the ELLIS units, together with Matthias, Betke, and Daniela. And I was just working hard on getting an ELLIS unit in Alicante, Spain, here started. But then the COVID-19 pandemic happened, and just reusing the previous slide, in case you haven't noticed, I happen to be in Spain, and Spain is this red line here, where you see our growth in the number of cases is pretty high, and the country is really struggling with how to combat the spread of the pandemic. I had worked in the past on epidemiological models using large-scale mobile data, because I have been working in telecommunications companies as a research director since the end of 2007. So I had a little bit of a deja vu, because I realized that data wasn't being leveraged to help us make better decisions, in the same way as it wasn't used in the Ebola outbreaks, or it wasn't used in the H1N1 flu pandemic. So I really thought about this, and I decided to take action. I wrote an article for one of the largest newspapers in Spain, and I contacted both my contacts in governments at various levels, and also my contacts, my old colleagues in the telco companies. And I sort of like tried to put together a team doing data science for the government in Valencia. So why to do that? Because we want to move to policy making situation where the decisions are made based on evidence, and the hypothesis is that the data is a reflection, of this evidence of an underlying reality. The data science team that we have created is a pilot for Spain, and the plan is to expand the methodology and the type of work that we do to the rest of the country. Our work was presented by, sorry, this is in Spanish, it was presented by the vice president, one of the vice presidents in Spain. We have a lot of vice presidents in Spain, Nadia Calviño, in a press release on the 27th of March. So why create such a team? As I told you, I've had a lot of experience working on infectious diseases and on the use of data for infectious diseases. And for many, many years, we've experienced a number of barriers that are preventing these technologies or these methodologies to be used in practical terms. So just a few days ago, a group of international scientists and practitioners, as you see on the slide, we published this paper where we highlight some of the key barriers that we have experienced and that prevent public administrations and governments from effectively using data to make better decisions. Because one of the main realities that the COVID-19 crisis has revealed is that decisions are being made without having evidence that this is the best, decision to make, or having partial, data. So why? One reason is because in public administrations and in governments, there is a lot of barriers related to capacity, to awareness, and to having a digital mindset. A lot of the public workers, they are not experts in data analysis. They don't have the digital skills necessary to be able to leverage, all this data. There is a barrier on accessing data, obviously, but there is also a lot of data available that could be useful. There might be certain concerns about privacy and data protection, but all the work that we are doing is fully aggregated analysis on anonymized data. So these concerns are actually really small. There is a big gap between the research community and the policy making and the decision making community. And there is also a gap in the maturity of the projects. So, now is not time to do research. It's not, at least if you are in an operational team, it's not time to come out with something, what if I do this or what if I do that? We have to deliver results every day by 5 p.m. So we don't have a lot of time to be experimenting. And then we've also realized that there tends to be a lack of preparedness for immediate organized action. And there seems to be all these, disorganized, decentralized, different ministries making different decisions in a not super coordinated, way. So what we've created is a data science organization, so to say, that is working directly with the president of the region. And I have it structured in 3 different teams. So we have a team doing mobile data analysis, which is aggregated mobile data coming from the mobile network, but also data possibly coming from other sources, like there are some companies that have mobile apps and they are willing to share aggregate data from their apps for the COVID-19 crisis. Then we have a second team that is working on epidemiological models that are able to build predictions on the number of infections with different levels of granularity, and both meta-population compartmental models like SAIR or individual models. We have to produce predictions every day on the number of expected cases, hospitalizations, intensive care units, and deaths, but also predictions on how long the growth is going to last until we reach the peak, and what is the impact of the different mobility contention measures on the progression of the disease. And then there is a third team that is doing all sorts of additional modeling that is neither modeling mobility nor modeling epidemiology. We have different data sources. We have some health data. To date, all the health data is pretty very aggregated and not super sensitive. We have the mobile data, and then we have other data sources, as I mentioned. In Spain, this weekend, as a result of this activity, and obviously, other people might be also thinking on similar lines, and the European Commission is also pushing for this. They passed a regulation on Saturday by which the 3 largest mobile operators are sort of like mandated to share aggregated mobility matrices with the Spanish National Office of Statistics, which is the one that is going to be sharing it with us. So there is actually, they made it like a legal requirement to do this. So the teams, they have very similar skills. Everyone has a lot of expertise on data science, machine learning, applied like to data. But then depending on the flavor of the team, there are some additional areas. So the mobile data team is also building a lot of visualizations of this really large scale data. The epidemiological team needs to know about epidemiological models, and then the generic model team is more of like a generic machine learning, kind of team. So what questions are we trying to answer? So on the mobility space, we are trying to understand how has the mobility changed because of the contention measures? Have the interventions impacted mobility? What type of mobility has been impacted? The long distance, the short distance, the inter-province, the within municipality, and also, is that enough? Where should mobility, drop further? From a policy-making perspective, the spatial granularity that is the most meaningful in Spain, or at least in this region of Spain, is what is called health zones and health departments, because those are the areas of competence or the way the healthcare system is structured in the region. So we are doing all the analysis at that level. I just wanted to show you a very quick visualization of some of the type of data that we're dealing with on the mobility space, and sort of like how you can see the different mobility between the different cities and parts of the city. And we can do that at the zip code level or municipality level. The epidemiological team is doing relatively standard epidemiological work. So we want to be able to predict how many infected people there are going to be under different mobility scenarios. We're going to be able to determine whether the social contention measures are enough or not. And also we're trying to maybe even infer what are the main causes of infection. And this will bring me to one of the actions that we launched this weekend. And then the additional modeling team is doing other types of predictions, like, I don't know, predicting deaths or predicting intensive care, looking for new data sources for anything that could be helpful to help policymakers make better decisions. We have a meeting every day at 9:30 where a member of the president's office comes, the data protection agency comes, and all the data scientists in the team come. And we do that every day. So we work really, really closely, both with the data protection agency and also with the actual policymakers that are making decisions, about this. Even if we have all this data, we realized that there was data that we didn't have, and there were questions that we couldn't answer. For example, we didn't have any data about the social contact behavior during the contention measures. So how are people getting infected? What kind of behavior are people having during the social contention? Are people leaving the home? What are they doing when they leave the home? Do they know who infected them if they are infected? Do they know anyone that has been infected? Have they been in close contact with them? So all these questions, we didn't have any data about that. We also didn't have any data on the economic and labor impact of the COVID-19 crisis on people's lives. We didn't know exactly what the prevalence of symptoms is in the population. I think the million-dollar question is how many people are really infected, because there are so many asymptomatic or slightly symptomatic people, and there are such few tests that the reported number of cases is really, really low when compared to the real number of cases. I don't know if you read the paper published a couple of days ago by the Imperial College COVID-19 Task Force, but that was one of the conclusions which was evident, for all the ones that were working on this. So we created this survey. I invite you to go check it out. We did that last week, and we decided to launch it in the weekend because we anticipated that new confinement measures were going to be deployed on Monday, which were. The survey is available in multiple languages. It's an anonymous, fully anonymous 24-question survey. We worked really hard to get it live before Monday, and we did that on Saturday at 8 p.m. Something completely unexpected to me happened, which was the survey became extremely viral. The initial deployment was in Google Forms. We kind of even collapsed Google Forms. In 36 hours, we collected 142,000 answers. I've never seen anything like this. The level of solidarity of the people and the engagement of all the people has been incredible. So Bernhard mentioned the fact that everything is going very fast and the cycle is very, very fast. I can tell you as an example this, we deployed it on Saturday, and we've been working constantly to write the results of the survey, and we just finished the paper this morning, where we describe everything that people have told us, but also we draw 10 conclusions or 10 implications from the answers from people. I cannot go into detail, but if you are interested, I can send you the paper, and I'd love to hear your feedback about it. As I mentioned, the way of working is very intense. We meet every day at 9:30, and we have to have results by 5 p.m. every day. We have a common code repository. Everyone has signed, obviously, NDAs and code of ethics. We have very strict data access controls for some of the data. So not all the teams have access to all the data, because if they don't need to, why would they have access to that? And we deal with all the online team communication using Slack. And that's all I have time to share with you. So thank you so much. I encourage you to fill out the survey and to advertise it. It's a worldwide survey right now, and I'll be happy to take any questions or comments that you might have. Thank you.
Thank you very much, Nuria. This was great. So our plan was that we have a question time at the end of each session. So if Richard Neher is on the line, we would now continue with Richard. And maybe while he connects and shares his first slide, I will briefly introduce him. So Richard Neher is a professor at the Biozentrum at the University of Basel in Switzerland, who's very active in the field of bacterial and viral evolution and modeling. He's trained as a physicist, and I originally got to know him when he was an independent Max Planck research group leader at our neighboring institute, the Max Planck Institute for Developmental Biology, and talked to him a number of times. He is one of these rare people who really connect a profound knowledge of physics and modeling with a deep understanding of the biology. And I think this puts him in a rather unique position now, and he's been very active in Switzerland and beyond, not only in driving the development of the field, but also in shaping public opinion and keeping us all on track and making sure that we all state our assumptions correctly, etc. So I'm very much looking forward to your presentation, Richard.
Yeah, thank you very much. It's a pleasure to share with you guys what we've been doing. And yeah, so what I was hoping to do over the next few minutes is tell you about two projects. One that has been going on for a long time and now sort of got into the limelight in the context of COVID-19. And then another one which we sort of stumbled into 4 weeks ago and has been keeping us busy quite a lot. You know, as Nuria just told you, we also got completely steamrolled by this outbreak. I sort of remember 8 weeks ago when we were pondering over the numbers in China. It seemed like a very, very remote event that's unfolding there, and we were sort of looking at Singapore whether something happened or not. But this has sort of very profoundly changed, and yeah, sort of we got completely sort of washed up in this between media attention and sort of trying to communicate to the public in some sensible way, doing policy, advising, and sort of doing research and developing tools at the same time has been a real challenge. But so far so good. So the one project that I was going to tell you about is NextStrain, which some of you might have heard about, and that is sort of meant to track the genome and genetic diversity of an outbreak of a pathogen. And this was initially, so it's an initiative that me and Trevor Bedford, a colleague in Seattle, started about 5 years ago now, mainly to study evolution of influenza. Influenza was sort of our prime target at the time, because this is a virus that sort of continuously circulates and where you need to update vaccines every week, every year or so. And hence it is very important to have an up-to-date picture of the genetic diversity. So when the first genomes of SARS-CoV-2 or HCoV came out in the mid of January in this year, we started to set up a small little NextStrain site for this so that you can see these genomes in the context of other genomes. And by the end of January, this became, or sort of mid-February, this became a huge operation, where suddenly the number of clicks that we had on NextStrain went from a few, about 1,000 a day to 100,000 a day or more. And suddenly we got a bill from Mapbox over a couple of $1,000s, which they nicely waived. But let me just tell you a few things about NextStrain. So if you go on NextStrain right now, I don't know whether this the Zoom thing works well enough to have a sort of full high-res screen, but let me know if you can't see what I'm telling you about. So what you see here is sort of a phylogenetic tree on the left of pretty much all available SARS-CoV-2 genomes to date. This is 2,500, and many of them really just came in the last 2-3 weeks. So the number of genomes that we have to analyze is really growing exponentially. To those that are not accustomed to looking at these kind of phylogenetic trees, what you have here at the very left, that is sort of essentially the genome of viruses that circulated at the very beginning of the outbreak. So the color blue here or purple is China. So in these viruses here, those are going to be many of them. Let me just increase the size of this a little bit. These are going to be the sort of the earliest genomes that were sampled. And as we now move to the right, we observe that these viruses, they accumulate mutations. So this is just random copying mistakes that the virus replication machinery makes as we go along. So these viruses here, they have sort of one mutation relative to the original Wuhan strain. Then these have two, and so forth. The color here on this graph currently, that is geographic location. So the red ones are from the US, the green, yellow ones are from Europe, and the bluish ones are from Asia. So we can now change this display here slightly to have this x-axis, not number of mutations, but sampling time. And then you get a sort of a picture like this, where sort of these recent genomes here, this is sort of these are genomes from just last week, right? This is sort of March 25, a genome from England. These red ones is sort of a big cluster of genomes from Washington, US. And the early Wuhan genomes, those would be from the beginning of the year or end of last year, where there's a few samples available sort of around Christmas last year from Wuhan. And so the root of the tree, that's sort of the presumed ancestor of this outbreak, that must have been at some point sort of late last year, probably in November. So our current best estimate here is something like late November, early December. And that is sort of only the ancestor of genetic diversity, not necessarily the ancestor of the entire outbreak. Yeah, so then you can sort of, for example, zoom in a little bit, and I prepared this here on a separate graph. So what you're looking at here now is only genomes from Iceland. And the one thing that, for example, it tells you, so Iceland is because they just like last week dumped 300 genomes onto the database. And what is remarkable here that Iceland at the time had probably like 500 or 800 cases, and they basically sequenced almost every single one of them, but like half of them. And what you see here is that these Icelandic genomes, these are all these dots, they are scattered across the tree. So you have, this clade here, you have lots of these Icelandic genomes, and there's one group of Icelandic genomes here, and sort of a bunch more here, which is basically telling you that these Icelandic genomes are completely, pretty much representative sample of European and US diversity. So this group here could actually be traced back to travel returnees from the US. So they got infected in the US and brought this virus back to Iceland. Most of the other ones had some degree of travel history to Europe, be it so skiing holidays, things like that. But then you also observe some outbreaks which sort of within Iceland, where you basically have exclusively Icelandic sequences that group together without any other sequences, suggesting that this was an introduction into Iceland, probably from a tourist that came back from Europe, and then sort of spread locally in little clusters. So that is just to illustrate that these genomes, what they allow you to do is gives these outbreaks structure, right? Sort of without these genomes, Iceland would just be, one number, 800 cases, something like this. But with these genomes, we know that, this is really like 10, 20 different little outbreaks that all got transported to Iceland by airplane, sort of people that got infected abroad, brought it back, and then it initiated local spread. So these genomes, they provide you sort of additional structure to the otherwise completely aggregated numbers that you get from just counting cases or counting dead people and things like that. Yeah, so that was a very brief overview here. Sort of I guess that's sort of most of this I said. So the resolution, the temporal resolution that we have here is about 2, 3, 4 weeks, and that is because we expect to see about one change on the tree every 2 weeks or so. This is quite stochastic. They are over dispersed, so they're not Poisson distributed. So sometimes you observe sort of 3 at once, and sometimes there's none for like 6 weeks. But this is sort of an intrinsic limit to the resolution at which you can differentiate different introductions and transmission chains. But another thing that one has to keep in mind that the overall picture that you get is quite heavily affected by the sampling and sequencing efforts, and Iceland being sort of a very specific example here that sequenced a lot. While if you did the same thing for Iran, you would end up empty-handed, because while they have a lot of cases, they didn't so far sequence anything.
Richard, we're getting some comments that you should move closer to the microphone.
Oh, sorry. Yeah, happy to do that. One sort of word of caution, while this might seem like a lot of genomes, it really isn't big data in any sense of the world, because nothing here is sort of independently and identically distributed, right? So total amount of information in 3,000 genomes is probably like 100 bits or so. So there isn't all that. This is not like big data in the classical sense. So just as a word of caution, if one sees a file with 100 megabases or a gigabyte, that does not mean that there is actually. This compresses pretty well. It's pretty small data for all practical purposes. Okay, then the other project that we stumbled into, and that really just started 4 weeks ago, pretty much exactly 4 weeks ago, when a colleague of mine in Sweden called me and told me, well, we really don't know what's coming. Can you give us some idea, some modeling, some prediction, some scenarios, whatever, that could help us to plan hospital capacity? And I told him, yeah, well, we kind of try to do something, but we'll be operating on sort of very, very sketchy data for the time being. But anyway, this developed into a sort of little web app where you can adjust all sorts of parameters of the classical epidemiological model that's called an SIR model with a whole bunch of different bells and whistles. Like we have this for different age groups, and there's sort of various different compartments for critically ill, sort of severely ill, and so forth. It's sort of a little bit SIR model on steroids, but basically it's a simple SIR model, a system of coupled ODEs that is solved forward in time. And what it allows you to do is sort of specify a particular population with an age distribution. Age is important because old people tend to be much more severely affected by this than younger people. You can specify how you think different mitigation measures or sort of infection control measures reduce the spread. And then you get sort of an output like this, where you have a model output sort of overlaid with sort of the actual observed cases. So this is data from Germany up until the day before yesterday, where they had 455 deaths. And you see that you can't possibly fit all these curves because all these the cases and the deaths and so forth, they all have sort of underreporting to various degrees. So we think that in most places, probably the cases are underreported by a factor of 5 to 50. Deaths are probably underreported by a factor of 2-ish. Depends again heavily on the place you look at. And then, of course, any prediction going forward is heavily dependent on what kind of measures are being implemented and how they are complied with. But this is just meant to give you some idea of how what we know about the virus in terms of severity and epidemiological parameters, and the sort of mitigation measures that were put in place, how they might conspire to generate different future dynamics. So it's quite explicitly not a prediction, but sort of a scenario, in that you make assumptions on what's going on, and then you see how they play out in this model. Yeah, sort of feel free to play around with this. This will going to change quite a bit over the next couple of days. We have sort of a new hosting setup and all sorts of things, but the basic idea and the link right now, which is just nealab.org/COVID-19, that will continue to work. How is this? Yeah, I'm happy to take questions, and yeah, wish you all the best with whatever is being planned.
Thank you very much, Richard. This was great. So a lot of questions have accumulated, and both to Richard and to Nuya. And I think we have about, good thing is we have more than 10 minutes to go through questions. So I think we'll be able to address quite a few. So maybe we start with Richard, or we can also mix things a little bit. So one question was, how fast does the virus mutate in comparison to pathogens that mutate too fast to vaccinate against effectively, e.g. HIV?
Yeah, so so whether something is mutating fast or not doesn't have an immediate bearing on whether it is a vaccinatable disease or not. So measles mutates about as fast as most other RNA viruses, and there's a perfectly good vaccine. HIV and flu have a similar mutation rate, and vaccination doesn't really work. So for HIV, not at all. For flu, it's sort of a very transient endeavor. And that has more to do with how the virus and the immune system interacts, and what sort of immune evasion mechanisms the virus has up its sleeve. So with flu, it's sort of a constantly changing glycan shield and sort of a whole bunch of decoy sites that can change quickly, but tend to be the immunodominant epitopes. So I'm not too concerned that vaccination here would not work. I think a vaccine has good potential to work, and certainly for an extended period of time. This could be that sort of 3-4 years down the road we see immune evasion, but for the time being, I don't think that's sort of a major concern. The mutation rate of the coronaviruses tends to be a little bit lower than most RNA viruses, So as I said, so about 2 mutations per month per genome, which is about a factor of 3 lower than some other RNA viruses.
So next question, and I should say, I don't see who asked the question, but whoever did, please send me an email because we're also working on this. So the question is, are you or others developing analytical tools integrating viral strain genomes with personal geolocalization data, for instance, from donated cell phone geolocalization history?
So the only information that we have on geolocalization is whatever was attached to the sample. I would think that the genetic resolution, so the genetic data gives you a very coarse relationship between different samples. That sort of can, I'm not quite sure how this would interact with sort of very precise geolocalization data. What we do have is some reports on travel history, which of course could be pulled from cell phone data, but there is no direct evidence that or direct connection that we use as of today.
I mean, I guess maybe the question is pointing at the possibility that if you know exactly what virus strain you find in which person, then that tells you something about the transmission history. And this way we might learn more about transmission and how to work against transmission.
Yeah, well, it will certainly tell you, it gives you pretty good information about from where the virus got introduced in the population. So whether, for example, this Iceland example that I showed, there's a bunch of Icelandic genomes that nest within a US clade. And that is pretty strong evidence that, yes, there was probably some travel returnee from the US to Iceland that brought the virus with.
So next question, I guess you get this question a lot. People worry about these kind of issues naturally, I think everybody. And this question is, how many of the different genomes can be considered different strains? And have you looked into associations to disease severity?
There have been claims to this effect that there is 2 different groups and one is more severe than the others. As far as I can tell, these claims are complete bogus. Because they usually compare samples that have completely different statistical properties. So if you compare a sample from early in the outbreak, where most of the samples were from sort of severely ill people in Wuhan, you get and compare this to a sample made later in a different context, you will get a lot higher mortality in that early sample than the later, but not because the virus is different, but the sampling strategies are different. So I don't think there's any evidence right now that mutations that we see have a functional effect. That doesn't mean that is not possible, but jumping to conclusions here is tempting, but not terribly well evidenced.
So maybe we give you a minute of a break and go to Nuria a little bit. So I think there were a number of comments to Nuria of people who want to contribute, both from inside Spain and outside, people asking for the link to your paper and people asking for the survey. So I think we can provide that subsequently on our collection of material. People are also asking about people are mentioning that the survey contains some countries but not others. So whether you're planning to include more countries.
Yes, definitely. And if anyone is willing to translate it, it's very simple. I send you Excel and you just translate the Excel to your language and automatically it is in that language. So we are doing everything we can, but there's a lot of countries in the world. So this is really a collaborative effort. I am speechless about the contributions of people. There's this concept of citizen science and engaging with citizens to inspire them into contributing, to make better science. And this response to the survey has been incredible. So I'd be more than happy to expand it to whatever languages you guys are interested in your countries. And and I will share the paper. Is almost done, but I've never written a paper so fast, I guess, in my life, like in one day, from nothing to having something. and also the methodology and all the analysis that we are doing and that we have done.
So next question, and I think that fits quite well with your general interests of doing machine learning with humans in the loop and interactions between machine learning and society. So this question is, how well are we able to predict the spread of the virus given so many dynamically changing parameters in terms of changing government regulations, changing mobility patterns, changes in medical capacity and so on? And how do we collect data in this scenario, considering that data collected on day 1 has presumably become redundant on day 20 of the outbreak?
Yeah, so that's a very good question. Every day we build 3 different models, which are meant to be like an upper bound, lower bound, and ideally the likely middle between them. And the models are, one of them is based on exponential growth. The other one is based on a SAIR model that was just explained in more detail. I didn't explain it because I anticipated that someone else was going to explain it in later presentations. And that SAIR model includes mobility, so you can adapt the rate of growth of infected depending on the mobility. And then we have a different model that is actually using a completely different approach. It's just a predictive model based on the time series of hospitalizations. Hospitalizations are a relatively good signal because everyone is in the hospital and they are get accounted for. And we know the rate of hospitalizations from infected. So we built a model using hospitalizations data to predict how many infected and how many hospitalized and it will be the next day. We also build models on intensive care and deaths. Having said this, the data is very noisy because the evidence depends on the protocol that is applied to gather that evidence. In most countries that I know of, they have changed the protocols multiple times throughout the crisis, and that has made a jump in the number of cases suddenly. Not because suddenly there are many more infected people. It's just because they are measuring them in a different way. They are testing more, they're testing less, they're focusing on testing this group versus this other group, you know, etc. So in the SAIR models that we build, the underlying model has a very high number of infected. And then we have another parameter that we infer, which is the detection rate. And this detection rate changes over time depending on the protocol that is being applied. So the model changes because the mobility might change, but not because of the detection rate changing. The number of infected is the number of infected. It's not changing because we decide to look at them or not. They exist. What we change is the parameter that is telling us from all the infected, how many are going to get accounted for. And this we also adapt every day depending on what the protocols are. For example, another challenge is the availability of tests. So even if they don't change the protocol, there are many regions that run out of tests. So suddenly they don't have any cases, like the number of cases that's like really slow because they don't have any tests to confirm, so they don't count them as official, you know, infected. So all these has to be taken into account in the model. Otherwise, the model doesn't explain reality. I mean, the model would totally fail. So it's it is a complicated process where there is an underlying mathematical model, but there is a lot of context, knowing the context, knowing what's going on, and tuning the parameters so they explain, you know, reality. The most inspiring and empowering thing here for me and for all the people in the team, we are all volunteers. We are working, I don't know, really, really hard every day, Saturday, Sunday, every day. And definitely the most inspiring is to feel the impact of the work, because every day it gets shared with the president and every day it is used for making decisions. And this is, I think, an incredible opportunity to show how civilians can work hand in hand with public officials and decision makers to try to, you know, make better decisions.
So you'll be happy to hear that we already have a few comments online of people volunteering to translate into other languages. I would like to try also. We have 2 minutes left, and since we have a number of people who are online in the Zoom conference, let's try some live questions also. So if I don't know if this is going to work, but as I mentioned, this is an experiment. So someone with a live questions to Richard or Nuria.
I actually have a question for Richard. Do you guys have any sense on immunization after you contract the disease? I know this is a somewhat debated topic, whether you would get immunized or not if you have gotten the coronavirus. Do you have? Do you know any? Can you share with us your views on that?
Yeah, so I fully agree. It's sort of an unsettled and somewhat controversial topic. There have been reports of reinfection. I don't really give too much on these because I think there are more reports even on sort of long-term viral shedding, and that PCR sort of comes up negative a few times and then positive again because there's some sort of viral RNA that's still being excreted. It's not so surprising. We do know that seroconversion does happen within 2 weeks or so. So people do mount an immune response and have IgM and IgG antibodies afterwards. What's probably most important for preventing infections would be IgAs. So yes, I do think there is going to be immunity, as with pretty much all other viruses of that type.
Okay. Oh, that's good
SARS
news.
too, we have seen sort of pretty good titers for at least a year. So I think these reports of reinfection are somewhat outliers.
All right. Oh, this is really great news. Otherwise, I think we're going to be really in bad shape once we reach this peak, because there will be another one if everyone is susceptible again.
Yeah, and while sort of I might expect that reinfection could happen after a couple of years when the virus changes, I would also expect that reinfection is much less severe, more like the stuff that we see in children now.
Cool.
So you have human coronaviruses that are just going around seasonally and cause cold, right? And it's quite how severe such an infection is depends a lot on your immunological predisposition. So I don't think it will be that bad going forward. Immunity is going to build up.
So I think we're perfectly on time to move to the next session, and I'll hand over to the session chair who is going to introduce our next speaker. Thank you very much.
Thank you very much.
Thank you. Bye. Thank you.
Thanks to Nuria and Richard especially.
Hi, everybody. My name is Gunnar Wert, and I'm happy to introduce the next speaker. So our next talk is by Christopher Mason. He's from the Weill Cornell Medical College in New York City. Chris is very much involved in the COVID-19 response and research in New York. It's great that we can get him on short notice and for some time here to share some data and some stories and some results related to COVID-19 in New York City and around the world. I think it's fair to say that Chris is a technologist, and he's driving and is being driven by technology, specifically on RNA and DNA sequencing technologies. So today he's going to talk about his recent efforts in global mapping of the RNA viruses, their hosts and their variants. Chris, I'm very glad you could make it. I'm really looking very much forward to your talk. So let me unmute. Chris, are you there?
Okay, great. Yes. Okay. Can you hear me okay?
Yes. We don't see your slides yet.
Yeah, so good morning and good afternoon, everyone. Thanks for having me. It's a pleasure to be here. As most of you know, we're in the middle of sort of the epicenter of the crisis in the US, and so I haven't been sleeping much. So if I sound sleepy, it's because I am sleepy. But let me share my screen here and go over some of the work that we're doing, and also some exciting new results I think that'll help everyone on some ideas and planning. So let me get this up here. So, I'm going to go, let me see if I can minimize this, and you can see my screen, right? Make sure it looks okay. Great. So I'll keep it brief. It's really just about a 10-minute talk just to go over some things we're doing in the lab and elsewhere around the city, and also from a global perspective, what we've been doing to look at the question of where the viruses might live elsewhere outside the body. So I'll start, obviously with the background. Most people know this this map. If you haven't been seeing it, it's a really great but yet terrifying map of where the cases are. And so things have really spiked here in the US recently. And you can see here there's many cases will almost certainly surpass 1 million probably within the next couple of days. So it's not news to anyone. But what we've been doing is both in the clinical samples and also in environmental sampling, this idea of can we see how many of the viruses and evidence of them in the environment, and then compare that to what we're finding in patients. This is kind of building off a consortium of which Gunnar and I have been working on for years. Actually, is a consortium of metagenomics and of researchers looking at subways in urban biomes as metasub. It's basically, we started sampling on March 16th to look in public surfaces and hospitals to really address the question empirically of is the virus surviving on surfaces. There's been a few papers looking in hospitals and other environments to see that they can sometimes get a virus that's there, and sometimes even infectious virus, which are two important distinct sample types. But we wanted to build this as a global map and see this as the pandemic progressed, what can we observe of what's present in the cities, and then also what we find in patients. So in particular, this work, Andre Callas, so I don't know if he's on the call, but has also been building basically a global KMER index, some of which you can browse at this website below to see what we see. We've been building this at the DNA side, and in the past few months, we started to build this index out also for the RNA side, with the idea that if we had been doing this routinely, we might have been able to actually track the emergence of different RNA viruses, as we already can for DNA viruses before it would have become potentially problematic. And so this is an infrastructure that we've been building on for years to begin this mapping. I'll share some of the results with you today. But this is kind of the concept and a very nice figure that Andre made to show this sort of concept. So there's a multi-sample sequence index, and you can search actually on this website below. Even if you don't know what a piece of DNA is, you can at least tell where it's being seen, which is something we'll end up need to be doing for other viruses that are constantly emerging. And also, since I know it's a machine learning group, we wanted to tell you one other bit of work from the consortium. This paper is online in a preprint, is predictive microbial signatures, just looking at DNA. Can you actually predict what city you're from and how much in different cities, can you predict, some more than others? Zurich and New York City are among the more mysterious cities, if you will, at least in terms of prediction accuracy, but population and surface type do often give you some sort of signatures that can use that the classifier can anchor onto. And we can get up to about 88% accuracy. If you give me a sample, we can likely tell you where in the world it came from at the DNA level. So this is some ongoing work with Metasub, and I actually didn't put the preprint location on here, but if you Google Metasub bioarchive, you'll probably be the first hit, almost certainly. And so that's the bit of the background of the consortium. And other things we've been doing about the consortium is they're very dedicated researchers and creative. You can see here, Alina, 1 year was detained a little while, and she said, well, can I sample this detention center as part of the project? So that became part of the sampling collection last year. Also, Naranhan Najrahan actually has got very creative with the people in labs you can see here in Singapore. So it's a very fun group, a lot of dedicated researchers. But in the past really few months, we've ramped up to do first a pilot sampling across these cities for looking at, historically we'd only looked at DNA, but now we're extracting DNA and RNA from every single sample to look at what's present. These are the first cities that started. And as of now, as of today, there's about 59 cities sampling about 10,000 locations across the world, as you can see on that map. So beginning this virus tracking to see what can we find in the different environments. So in particular, you can see, for example, this is a picture in Seattle, Washington. You can see here's David Denko from the lab sampling in Penn Station. You can see here in Alexanderplatz in Berlin. This is in Buenos Aires, the sampling location, some of the areas. This is in Daegu in South Korea, where there was a really big outbreak there a few weeks ago. We were sampling there, and also in Brazil and Sao Paulo, and then also in Tokyo, Japan, among other places. This gives you ideas of, as the sampling's happening, there's an online app where you can see the geotag and the picture of the location. So one interesting bit of data of what we've seen so far from the shotgun sequencing of public surfaces is about 10% of the RNA is human, about 1 to 2% of it's viral, and a good amount's bacteria. But if you rank the viruses that we've just been seeing, and this is very new data, a couple of days old, we so far have not seen coronavirus, which is a bit of good news, but we've seen influenza, we've seen pneumonoviruses, we've seen other sort of phage viruses. There are a lot of viruses to be found all around us. Some of these things are not spread. This is just the average abundance across the first 14 samples we sequenced. And you can see, number of reads, we're seeing a good number of reads and even coverage across the genome. So this is very new data, but it's interesting to see what else is out there in the world around us. And we just had another 60 coming off the sequencer today. So we'll share this data within the Metasub consortium, and anyone on this call is welcome to join the calls or hop on and play with the data as well. So that's a bit of a shotgun sequencing of surfaces. Two other updates I want to give you quickly about what we're doing is ways to make this faster. So in particular, we're working on a protocol called LAMP, which is a loop-mediated isothermal amplification. The way it works is every time a polymerase incorporates a base, the pH starts to drop in the solution. So as the reaction proceeds, it'll drop the pH and change the color. So it's basically just a pH test. And so using a synthetic RNA, you can see here at 0 minutes, it's all pink. Once it's at 20 minutes, you can see the color starts to change, especially for high viral abundance. And then once it's finished at 30 or 40 minutes, we've gotten it down to 30. We can see between 10 to 100 viral copies of what we can detect with this reaction. So We're excited about this because we've gotten it working as a rapid test in environmental samples, and we can confirm these negative detection of coronavirus or SARS-CoV-2. It works though for the clinical positives, and also the negative controls, it also works. So this is building off some work that was first testing it in China that showed that it was possible, and we've now done this on over 200 samples, and it looks really good, about 94% sensitivity and about 99% specificity. So we're very excited about this as a maybe more rapid test that we just got the IRB approved yesterday. We're going to be deploying this at the hospital here in New York City, hopefully by Friday, actually. So that's not everything we're doing. I also have kind of a dream slide of if we could do everything for every sample, which we can't always do because we're limited in material, but we're doing LAMP, we're also doing QRT-PCR and ribodepleted and sometimes poly-RNA-seq. We've begun doing some direct RNA sequencing and MinION work, and also doing look at capture panels with Twist, for example, with these samples. So this is kind of my dream slide. We're doing mostly these top 4 and sometimes doing a little bit in the bottom when we have enough RNA from different clinical samples. So speaking of, what we've seen for clinical samples so far is if you do a ribodepleted RNA-seq, this is a coverage plot of what we see. So you can actually get really good coverage of the whole genome. And we're seeing anywhere from 60 to 70% of the virus from nasopharyngeal swabs to as much lower, like 0.1% or lower. It really depends on the sample. Actually, I have a slide of the correlation between the two, which looks so far pretty good, which I'll show you in a second. But we've been able to get consensus alignment. And as most of you probably have been doing, we've been uploading all of our data into Nexttrain. So you can see some of our samples down here in red. This is actually from a plot from a couple weeks ago. But we begun uploading all the samples here, which is I recommend. If you upload them to GISAD, then Nexttrain pulls that data from there. I highly recommend this site. If you haven't been there, it's probably one of the best sites to look at. You can see it's here. I think my time is up almost at 10 minutes, but if you haven't been to the site, it does a really great video of the sort of history of the phylogeny and distribution of the samples around the world. So if there's a GISAD is essentially where people have been depositing their data, so we're putting our data. If you haven't already, any of you doing sequencing, even if you're doing targeted sequencing or shotgun sequencing for metatranscriptomics, you can do the alignments and call variants and then upload the FASTA file to GISAD. It's a really great resource that, at least as far as I know, is the most centralized location for all the viral sequences. So that's where we're putting all our data, and I recommend it everyone. So again, if you haven't seen this site, I presume most of you probably have, but if you haven't, it's really one of the more beautiful illustrations of when and how and where different viral strains have moved around the world. And also at the bottom, you can see where are the different phylogenetic distances, how are they mapping in terms of mutational frequency across the different strains that have been sequenced. So really extraordinary website. It's built by Trevor Bedford and his colleagues, and it's really fun to work with as well. So that's a quick update there. And then the last comments are we're trying to get set up pop-up labs across New York City to make it very easy to do this LAMP assay in the next basically couple of weeks. We're getting this set up hopefully soon. I think Danielle Bezin's on the call. We've done this before at other meetings where we just swab people's phones and sequence it and tell them what's there. It's just, in this case, it's for fun, but now we're trying to do this more for clinical purposes for testing people. And the idea of setting up pop-up labs all over New York City, we're working with the city council and also the mayor's office to begin to set this up, which we're working with right now with this LAMP assay, which we're submitting for FDA approval hopefully very shortly. So the long-term dream would be to not just look at the LAMP positive samples in RNA sequencing, but look at genetic variation, differential expression in the host cells that are there, look at epitope diversity, host immune cell diversity, look at changes in RNA function, and also anything else that's there. This is kind of my dream slide of everything we can do on the computational side with these samples from a host and microbial perspective. And then I'll close with a new initiative that just started on Monday, which is a host genetics initiative. So it's open to anyone to sign up. It's basically a place where people are looking at host susceptibility for sort of people that have high and low viral titers and sharing data from genome sequencing to array data to functional genomics data as well. That's my last plug for things. So stay tuned. Thanks to everyone. Thanks to Gunnar for the invitation and everyone else on the call. Thanks to especially the clinical collaborators and everyone in the Mason Lab. And thank you very much for your time. And also thank you, Krista, Ben, and the councilman for helping across setting up all the sampling around the world. And that's it.
Thanks, Chris. So I enabled questions for everybody on Zoom as participants. So you could just go ahead, unmute yourself, ask a question. So maybe I just ask one. So did you actually sample in the hospital in different areas, different maybe on the bed or indoors and so on, and try to see whether you find the virus in these COVID units?
Yeah, so we just started that sampling, so at least in New York City, but other cities have already been sampling. So one team in Bangkok has been doing it. The team in Singapore has started doing it. So we had to convince our IRB that it was non-human subjects material because they said, but won't you get some human DNA? And I said, we can, but we can also map those reads out. So we had to get a special IRB exemption. If anyone needs an email about how sampling public surfaces does not constitute human subjects research, I'm happy to send this to you. But so we just started that at Cornell, but other cities have already started.
And is it understood which surfaces are well carrying the virus? Do we have that information?
Yeah, actually, the best example came from, there's a preprint that shows this from Ong et al. It is here. I'll post this. It's a JAMA paper that's already come online. I'll post this in the message board, at least in terms of some of the rationale for doing the survey. Here, I'll share my screen again really quickly, but I posted the article. Is this paper just came out earlier last month, is looking at the air surface and sort of contamination by the virus. And what's interesting is they found little evidence of it in the air. Other papers have seen this possibly in the air, but they noted, there are basically 2 patients where they didn't seem to have that much viral shedding. But what really was extraordinary is this one patient had left it almost all over the room. So patient C, he found it basically 87% of the room sites, including toilet seats, outlet fans, and basically it was all over the room. Positive stool samples and also pretty reasonably high viral titer, you can see here. So this is a really interesting paper to look at the question of, this actually is a very strong rationale for why we should be looking and thinking about different cleaning methods, because it can be shed, it can show up in the environment in that one patient basically all over the room, which is kind of terrifying. But again, that's a virus present. They didn't test in that paper whether it would still be infectious, but they did see the virus everywhere, which means it probably was released for a little while.
Okay, so there's a question online. Is it possible to observe correlation between prevalence of old coronaviruses in the population or in the area and the severity of the SARS outbreak?
Oh, great question. So the beginning of that kind of correlation analysis, we've just been talking about in the meta subgroup and some of the discussions. So short answer is we don't know. That's a really fantastic question that I think as the world collects more data and keeps track of it, this will be the most quantified viral outbreak ever, which actually is awful, but also gives us a really important way to learn from it and plan for future outbreaks. So we don't know yet.
Yeah, so there's a second part of the question actually. So it might help answer the question if being infected to those common cold viruses improves our immune response.
Yeah, great. We have seen co-infectivity in one patient that had H1N1 plus SARS-CoV-2. So we have seen rarely some patients that have both, but it can happen for sure.
Yeah. So there's a technical question. Why is the direct RNA sequencing on MinION so slow, 1 to 3 days? So why does it take so long?
Oh, it's a good point. So it can be done within a day. It's just because the protocol, if you want to let the run finish, normally you take 48 hours. So you can do base calling right away and just put the data up. That is true. But normally you let the flow cells run for about 48 hours. That's the main. I'm just being I guess conservative with the number, but to be fair, you can sequence within 30 minutes or an hour on a MinION if you just stop the sequencing. That is very true. Yes.
Yeah. So are there other questions live in the Zoom meeting? Not at the moment. Richard, Bernard, Oliver, no.
Maybe a brief one. You just said you also see co-infections with influenza. Would you expect that that makes a significant difference for the clinical outcome?
At least in the patient we only have one, so it's a case report at this point. It did have more complications, but we just don't know at this point. We just updated the IRB so we can look at the electronic medical health records. We can track all the molecular data, the treatment data. All these questions are ongoing. Also, a lot of industry groups are treating patients and scanning those data. So I think it is, we don't know yet, but it seems like so far, yes, from just one case report, but we just don't know.
I have 2 more questions online. So in Germany, there's a Corona app in preparation developed by the Robert Koch Institute to identify infection chains. Data are anonymously collected. Is it foreseen or planned to use this kind of data also as input for your project, maybe for predictive analysis?
Yes, if the data is available. I mean, I think at this point we will we would do it for any data we can get our hands on, I'd say. And I think I'm sure you're the same way. So if we can get the data, we'll use it, basically. Yeah. It's actually, I've never seen the scientific community be more collaborative, not only within the MetaSub group, but just really globally. So it's actually very, although I'm not seeing my wife as much. So if anyone can tell her I still love her, that's my biggest problem right now. But other than that, really collaborative and wonderful. Yeah, and my daughter as well. So she's at home now. But yeah.
So speaking about collaborative, so how is it working data sharing wise? So you collect a lot of data now, you sequence a lot of data. I guess there may be some privacy concerns, but can you share this data with the broader community in machine learning so they can actually do something with it if they want to do it? I mean, we have 1,300 people online right now, so and they would look for data sets to look into.
Yes, and if there's a distribution list, all the subway samples, because they're environmental, we can share them really immediately. The patient samples, we're working with the Host Genetics Initiative and Cornell. I think eventually we can definitely share the viral sequences. Those will eventually become public. We just have to figure out a mechanism. So in the US, we use dbGaP, which is an atrociously difficult system to get data, but it does work and it maintains privacy. But we could put them also in the ENA or other, I mean, certainly I think it'd be very easy to quickly share the viral sequence, any non-human reads. I don't see any reason why we can not share them quickly. So we're working on that now. We have a Wasabi link for downloading the subway data, and that's just gone live this morning. So we can share that as well.
Yeah, it would be great. So there's another question, maybe more practical nature. Can COVID virus be transmitted via the packaging in supermarkets?
So we've been trying, it's possible, yes, although we have not demonstrated it, but I know when we get groceries delivered, we spray them down and let them sit for like 15 minutes out of an abundance of caution. So it would be, there has been enough other data showing that the virus can move through the air, that it can survive on surfaces and still be potentially infectious even after 9 days. So it is possible, but it is unlikely. It seems to survive less well on cloth and cardboard, but on plastic and metal, it does surprisingly well. But that's in the dark. If you put something outside, the UV radiation from the sun will degrade the virus within minutes, basically. So it is a wimpy virus outside of the human body, but if it's in a dark place and it's sitting there for a while, it is possible. But I don't want to be alarmist. But it has been shown that it's possible to survive.
Yeah. So there's another question more about prices for these tests, and specifically for the LAMP test. So maybe you can generally say something about prices for these tests.
Yeah, so RNA sequencing, most of you know, is in the range of $250 to $300 a sample for prep, and then sometimes more, sometimes less. But then, so you're looking at usually around $400 to $500 per sample for RNA sequencing. The LAMP assay is very cheap. RT-PCR is usually $50 a sample. LAMP is like $20 a sample, if you include everything. So the chemistry is only about $5 per sample. If you need some protective gear, if you're adding another pipette or reagents, labor, it could be no more than $20 per sample. So we think at scale it could be even cheaper, in the range of, I'd say, $10 a sample if you do a lot of them.
Yeah. Okay. Is there any question from the audience in Zoom? Any more? I have one more online question. So, is there any? It's relation with pneumonia and COVID. So is there any prevalent bacteria strain that is responsible for the pneumonia related COVID-19? Can one see that?
If it's from some of the samples, the pneumonia, if it's we're doing DNA and RNA sequencing. So if it's there, we could see it for the ones we've done both profiling on. But pneumonia, if it's the bacterial pneumonia, we'd have to get the DNA from it. Yeah. Unless it's expressing RNA, then you could see it as well. Potentially, yes. Potentially, you could just use RNA. Yeah.
Okay, any more questions? I think we're good. We are, I think, quite good on time. So maybe thanks a lot for giving your presentation.
Thank you all. Thank you. It's a pleasure to see you all. Good to see you again, Gunnar. I'm sure I'll see you soon. I'm sure see you all. I'll be in this room for the next 8 weeks. So if anyone needs anything, I'll be right here, basically.
Yeah, okay. Good to see you then.
I'm
so
great. Thanks.
So let's move on to the next. I think we are maybe 1 minute early or 2 minutes early. So, but I guess we can get started. Bernard, what do you think? So, yeah, okay. So I would like to hand off the session to the next chair.
That would be me. My name is Sepp Hochreiter. I'm fellow of the ELLIS Health program. I'm chairing the session about drug discovery using AI and machine learning for drug discovery. Our first speaker is from the Linz ELLIS unit. It's Günther Klambauer. He's also with Johannes Kepler University in Linz and is leading a research group on AI for drug discovery. And it's your floor, Günther.
Thanks, Sepp, for the introduction. So I'll try to share my screen. Okay, so I would like to thank, of course, the organizing committee for giving me the opportunity to speak here. I guess, as Sepp has already said, so we are an ELLIS unit in Linz, located in Linz, and I'm leading a group there on drug discovery projects. I call it AI and Life Science Group with a lot of people. And we had in the past and in the present, we always worked together with pharmaceutical. So only for the there will be about in the molecular scale. So we've heard a lot of things about patient scale, like tracking, also maybe microscopic or patient level or patient scale, things like healthcare management. And the speaker before me have already introduced genome sequencing, which would be targeting or doing relating ML and using ML and AI methods in the molecular scale. So there are multiple ways how you would approach this. So on the left-hand side, you see a schematic representation of the coronavirus. So these purple things and so on, these are proteins, structural proteins, for example, that the virus has. And in a drug discovery approach, you would try to design a small molecule, something like that, to inhibit or to hamper the virus, for example, to break some of its proteins or to inhibit some of the machinery that it needs to replicate and to spread. So there are multiple ways. So it has this coronavirus is a relatively simple thing with only a handful of proteins, but you still have a lot of ways how you could approach this to basically hamper the virus in a drug discovery approach. Or you could also think of antibodies. I will not focus this talk too much about this, but also antibodies could be designed to inhibit the protein, and that's basically what our body does. So how do we get to the computer-aided drug discovery then? A lot of work is currently in such a drug discovery project and process. A lot of work is done already on the computer. You see that there are a number of things listed. I will only detail two things, and very quickly I will talk about compound design, which can be done by machine learning, and also virtual screening. So machine learning can influence all these areas. I will show a bit in these two areas what it can be done, and then I will show an example project, what we have done. Okay, so compound design, there has been lately a lot of work since 2016 or so on using machine learning methods to generate molecules. So one of the first was an LSTM using generating compounds, a string representation of compounds, but there is now variational autoencoders generating molecules. There are GANs generating molecules, also reinforcement learning in combination with RNNs, for example, and even hybrid approaches. So overall, I think there are around 90 to 100 publications suggesting molecule generator with machine learning techniques. And these are very valuable strategies. However, I want to, so they can be, they can be a bit slow in the current crisis, because these molecules, they have to be still synthesized and so on. So it could be a bit faster if one does virtual screening, which is the second thing. So virtual screening is basically using a computer, using a model, machine learning model, to screen through a large database of molecules that are already available. So typically there are a lot of large databases around of molecules that are quickly available and can be purchased and tested in the range of millions or billions even. And there are two principled approaches, like ligand-based and structure-based approaches. Ligand-based approaches, they revolve around the ligand, which is the small molecule here that we want to design, and structure-based approaches. So these are computational techniques that basically use the 3D shape of the 3D coordinates, the 3D shape of the protein, and try to dock a small molecule, try to fit the small molecule in. Involves typically molecular dynamics situations. And of course, also here there are hybrid approaches. Again, I will focus here on the ligand-based thing. Of course, also in the structure-based area, machine learning can play a role. But here it's very clear how machine learning can be used in a ligand-based, because you typically have labeled molecules that can bind to a protein or that can inhibit the protein, and small molecules that do not have this property, and you can build supervised models, then perform virtual screening and select some compounds. Overall, this is a typical drug discovery pipeline, so it's a very famous figure. It can take very long, so it can take even up to a couple of years to finally develop a compound. So here you see a very long time scale. Of course, this can be much faster, but it's very typical that one starts with a large compound library with millions of compounds, then uses a virtual screening approach to select a compound library in the range of tens of thousands that are then slowly reduced to a couple of favorable candidates that go into clinical trials, which also take a long time. And then if you're lucky, you get one approved drug. So now with the current corona crisis, we have to speed this process up a lot, and here machine learning can help a lot, because if you select very good molecules at the beginning, you can proceed faster, and you have a lower chance that accurate machine models can already predict side effects or low efficacy and so on. So how does this present from a machine learning perspective? From the machine learning perspective, it's quite simple. So I'm presenting also a simplified view here. So your input are molecules, and you have labeled data, actives and inactives in the simple case, and you build a model. But what is the typical case that you do not want to predict or know the activity of a molecule in a certain biological effect, or whether binding to a single protein or not, but typically you want multiple effects at once. And here you can, for example, use multitask learning to have in a simple neural network with multiple output units that model multiple biological effects of a small molecule at once. And it's very typical that such label matrices like this here have a large percentage of missing labels. So this is very natural here that we don't have a lot of knowledge. This also represents our knowledge about small molecules, that the large part of the effects of small molecules we do not know. Also, from the machine learning perspective, molecules are very interesting from the representation view. So there are different ways how you can represent a small molecule. Of course, you can extract some features and represent it as a vector. The community calls this so-called fingerprint features. There is a string representation that I quickly already announced called the smiles representation, which goes neatly together with the RNNs. Of course, very natural, and we all learned it in school. It's a graph representation. Here, the graph neural networks could work, and there are also other representation, like with sets of fragments, and also representing this as a point cloud and using some point-based neural networks. So all of these representations can go with some machine learning techniques. Some years back, it was already shown that deep neural networks outperform other techniques at this task of predicting a drug target. So given a molecule, predict to which proteins it will bind. The gap was not as huge as for image recognition, but still it improved in all of the last years with attention mechanisms, and on this could be further improved. And commercial software at that point of time was far off at predicting drug targets. In another example here, for example, also toxic effects can be predicted quite well using a deep neural network with in a multitask setting, predicting multiple neural net toxic effects at once. So here would be toxic effects, and here would be the average thing, and deep neural network can do that very well. It's even the case that for some tasks, for some proteins, for some of the drug targets, the predictive quality is so high that a wet lab procedure can be replaced. So typically, you also are from partners from pharmaceutical companies say, if you reach an area in the ROC curve larger than 90 with a model, they consider this a virtual assay and put thinking about replacing a wet lab testing procedure with a computational technique or machine learning model in this case. Why can it work so well? Because there are typically large databases for learning. So for example, the PubChem database offers 270 million triplets. A triplet means a molecule measured for a particular biological target, a drug target, for example, a protein. Can this molecule inhibit this protein? And you have an activity value, which can be binary, but also regression tasks, of course, make sense. But overall, so PubChem and Campbell offer these activity data. DrugBank is a data set of marketed drugs, and there is a Sync database, which is a large, contains a large number of molecules. So assume you have a trained neural network, then those which select the molecules that can have potential toxic effects. Am I still hearable? Okay.
You were gone, but now we hear you again.
Switch off your video for now.
Okay. And I'm showing the result of such a study. So we applied this concept of virtual screening recently to coronavirus inhibitors. We were a bit lucky that we had a pre-trained network called that was trained on a large number of biological effects. So that data set comprising of Campbell, PubChem, and Sync. And so this network, the architecture is so-called SMILES LSTM. So it works on the SMILES representation and employs an LSTM, predicts multiple output at once in a multitask setting. And of these 600 different effects that it predicts, 4 were related to coronavirus. So there was some data present in public databases about the coronavirus, which is the old coronavirus SARS-CoV-1, and concretely on 2 very important proteins that the coronavirus needs for replication, so-called proteases. And we use this predictions to screen a database. And the good thing is that these 2 proteins are relatively conserved between SARS coronavirus 1 and 2. So we could relatively fast perform a virtual screening of the Sync database and report a list of molecules that could be potential inhibitors for the SARS coronavirus. We provided this list of the 30,000 top-ranked molecules via GitHub. I will share, the link will be at the end of the presentation. And so these molecules that I'm showing here could be potential inhibitors of the coronavirus and could be tested. The good thing about the Sync database is that it directly, so you can click on each molecule and find a vendor and relatively easily get this molecule purchased and tested. We also screened with this model, of course, DrugBank, and we got a couple of, we got a ranking of molecules that are already on the market, so marketed drugs that that would have the advantage that you can test these drugs faster in clinical phases because they're already on the market, so there could be a faster way to getting to drugs. These results on DrugBank can be requested from us, and we would share it for scientific purposes. My last point, I'm almost finished. So there will be a similar challenge announced very soon from a European initiative, so the JEDI initiative, and it's going to be the billion molecules against COVID-19 challenge. It's very similar to what I've shown. So teams like machine learners, groups with ours are asked to provide lists of molecules, asked to screen large databases for potential molecules that can inhibit this virus. They should have at least should be screened for at least 5 drug targets. So I showed results on 2 drug targets, and in 2 follow-up stages, these predictions from the different methods and teams will be tested experimentally and validated. This will be announced very soon. It's very short, so running only a couple of weeks, open to all teams in the world, and the prize money is over 2 million euro distributed across 3 challenges. So yeah, there's already some information. Please stay tuned. The challenge starts anytime soon. Okay, so just a quick wrap up. So I've shown how machine learning can help in this corona crisis in the area of drug discovery. Machine learning methods are already employed, but there's still a lot of room to improve. Large data sets would be available, and machine learning can especially help because these are quite fast methods. And I've shown a screening of a very simple model, pre-trained model, a screening of the Sync database and the drug bank database. So all of this information and also the slides will be on the Git. So the GitHub is already on. There is already also the small technical report on what we did, and of course the list of the molecules. And we linked a lot of database, and also of course this the data set that we can be used for training of these 4 assays related to the SARS coronavirus is uploaded there. Yes, so that's it from my side. I'm the first one to go over the time limit for Primer. Okay.
Okay. Thank you, Günther. Questions are at the end. I move on to the next talk. The next talk is by Joshua Banjo. He's one of the biggest shot in our community, Turing Award winner. But from my perspective, one of the sharpest brains I ever encountered. He's a very good guy. And now he's on our side to fight against the coronavirus. Joshua, it's your stage.
Thank you, Sepp, for these kind words. Let me try to share my screen. Wait, what is it? I don't understand what it's asking me.
It asks for permission.
Desktop 1, is that what I want?
Oh.
Yes. Shit. Sorry, guys. It's asking me to open my preferences. I thought this was already done.
Yeah, you have to allow the capture of your screen.
Yes.
You have to log out and come in.
So you will have to go out and come in again. So maybe as a step, you might want to take one question in the meantime.
That's a good idea.
We see already the slides.
Oh yeah, right.
Oh, okay. So let me start quickly. So I wanted to go over a few things that are going on at Mila. Actually, I'm going to talk mostly about 2 projects, and one is about drug discovery, but the other is more about contact tracing. Let me try to do a screen share. Do you see this? Do you see the screen properly?
Yeah, we see it. Yeah.
Okay. So I mean, I want to start by saying that I'm quite enthusiastic about the solidarity that I'm seeing in the scientific community in general, and in the machine learning community in particular. People are stopping what they usually do and forgetting about publications and egos and money and all the competition we usually have, and just focusing on saving lives. So at Mila, which has about 400 researchers, mostly grad students doing machine learning, many projects have started. There's also an effort to coordinate the different projects within Canada, organized by CFAR. This is a website for this. I guess the slides will be available later or something. Yep. So at Mila, we also have a webpage to list different projects. One of my postdocs contributed to a paper that's now an archive to try to survey the different machine learning methods that were published to try to fight COVID. And that was already like last week, but things are moving very fast. So covering things like diagnosis, drug discovery, AP models, social media. So I'm going to just talk about these two projects that we're working on, contact tracing and drug discovery. Sorry. Okay, so I want to mention a paper that just came out yesterday. Things are moving so fast, it's amazing. In Science, where they used a form of epidemiological modeling in order to demonstrate in simulation that if we can do digital tracing, contact tracing using phones, rather than what is commonly used in many countries, including what is going on currently in Canada, that is, hand tracing by, interviewing people and asking them who they met. You can actually have a very significant impact on the R0 of the disease, bringing it below 1. So I think this is a very important ingredient in the fight against COVID-19. So we are developing such an app. It's not just people at Milab. We're collaborating with people from around the world, people also in companies. And we're starting from the code base of MIT Media Lab that has an app called Safe Paths, where there are hundreds of developers that are contributing, and we are also adding our own contributions to this. The two particular things of the way that we're developing our app is on privacy and machine learning. So on the privacy side, I will explain that there are challenges because it's kind of it may seem like a contradiction to have both privacy and being able to protect, to intersect people's trajectories and to protect privacy and yeah, together. But it's really important for us in Canada because both the government and the public would not accept an app which doesn't match our sort of cultural expectations. And if we don't get a wide adoption, then the thing is not very useful. And I don't think the government will force people to use something like this. So it has to be something that people accept. On the machine learning side, the advantage of using machine learning is that in addition to just telling a person or their app that they were in contact, say 5 days ago with somebody who later tested positive, now you can combine a lot of pieces of more fuzzy information, like I've met a bunch of people, we don't know what their status is, or maybe I met somebody who met somebody who met somebody who was tested positive. What does that mean for my own risk? So that's where you need machine learning, right? But before I go there, let me go through a little bit of the privacy versus health. So from a ethical point of view, we really have two conflicting values that somehow we need to try to find a good compromise over. And I think that's where technology can be used. But there's still going to be a dilemma, and maybe the solution is going to be different in different countries, depending on how much you value privacy, for example. So for example, should governments have access to your full trajectory? In many countries, people would say no. We don't want people to know where you've been. We don't want people, we don't want a government or say the police to know who you've been meeting. Another ethical concern, which is important, is, and not maybe less obvious at first sight, is that if the information about who is high risk or infected becomes public information, or somebody that some information you could get if you're close to a person and somehow their phone tells your phone that they are a high risk, so that you can stay away. It might seem like a good thing, but then you might turn against that person and stigmatize them, and this question of human dignity here and human rights, which have to be balanced with the advantage of having information that helps people avoid risks, or even that deter people from acting irresponsibly. So think about if you knew that when you walk around the city, that others that cross path with you might know your risk level, and if you have a high risk, even if you're irresponsible but you don't want other people to reject you, you might decide that you stay at home and do what the government told you to do. But on the other hand, that goes against the privacy requirement that we don't want the infection status, infection risk of a person to be revealed to others. So we have these contradictions, and we have to understand them and then use technology to our best to try to find a good compromise. Now, one thing we are trying to do here in terms of strategy is start by deploying an app which will be very strong on privacy, and see how far we get in terms of reducing the R0, the rate of spread of the virus. Because there's a threshold value of 1, as many of you know. And so if we can combine with many other things that are going on in society, if we can get the R0 below 1, then you're basically done. And then we don't need to endanger democracy and privacy. And of course, if it doesn't work, then governments probably want to push more on the pedal of less privacy and stronger health protection. Okay, so one of the key ingredients that we're working on to try to find a good compromise is a set of algorithms that do what's called private set intersection. So what this is about is I have a set of points where I've been at time, location points, and you have a set of points, and would like to find the intersection of those points, but we don't want to have have access to each other's sets, right? So it sounds like it's impossible, but people in cryptography and so on have been working on these things, and there are solutions that are not necessarily perfect against all attacks, but they're pretty strong. And it turns out that we've been working with this group that just pushed out a paper maybe a couple of weeks before COVID became a big thing here. And you can find their paper online. It's called Contact Tracing Mobile Apps for COVID-19, Privacy Considerations and Related Trade-offs. So what's important in this method is that the trajectories stay on each person's phone, right? That's so we can have a sort of peer-to-peer exchange of information using the private set intersection algorithms. The ideal scenario is if we can get Bluetooth to work, but there are Bluetooth implementation issues. If you have Bluetooth, then you can guarantee that it's only the phones that were physically close to each other that are going to exchange this random key that could be generated when they're close to each other, that is going to later allow them to exchange information. So you need something like this, right? That we have a way to communicate after we've met, so that if I get infected and I know about it, I can tell you that information in an anonymous way. The general idea is we're going to convert this random key into a private mailbox sitting in a server, and only people who have this private key can either read or write from that mailbox. In addition, the way that that information is about, say, my location and so on is going to be converted into a mailbox ID can be split across multiple servers. And the idea of this is that even if some of the servers are compromised by some attacker, they would not be able to unlock those mailboxes. They would have to be all like colluding in order to have access to that information. Okay, so that's the privacy consideration that we are working on and implementing right now. The machine learning considerations. So what are we trying to do, first of all? So what you want is that each phone tries to predict the posterior probability that the person was infected for each of the days, say, in the last 2 weeks. 2 weeks because this is the time when you could have been infecting somebody. And you want that information because it's not my status now that matters for the person I met 6 days ago. It's the probability that I was infected 6 days ago. And so you want to keep that. You want to compute those posterior probabilities for all the past days. Say, I mean, you could do hours, but days is probably fine. So we can't afford to compute the joint probability of everybody's infection status. And I mean, that would, anyway, it's hard to represent that anyways. And because we are doing this peer-to-peer thing, we can only compute things locally. So, so we're going to have the different phones communicate with each other about their beliefs that they're being infected. But there's a chicken and egg problem, because my belief becomes an input. My belief that I was infected, say, 6 days ago, becomes an input for your calculation of your belief that you're infected now because you met me 6 days ago. And so we depend on each other's beliefs. Okay, and then the right way to think about this in machine learning is to think of the different phones exchanging information so that they can converge to a consistent estimation of the marginal distributions over all the phones of all these probabilities. And loopy belief propagation seems the right kind of algorithm to achieve this. So we are implementing these things. Now, within loopy belief propagation, you still need to estimate these conditional probabilities. And that's where we might use machine learning, or even simpler models, which we want to put on the phones before we get data, that are based on epidemiological model, epi data that are currently being collected from around the world. So these models would be very simple. They might be like naive Bayes models. But what's nice is we can put them on the phones right now. We can also use those models, and we have done that, so to build simulators. So the same way that epidemiologists are building simulators. So these models can then be used to generate training data for machine learning models. That training data could be used to pre-train our predictors before we get enough data from the real data collection of the app, which is going to take at least a week, maybe 2 weeks to get enough data because of the delay between things happening and test results. So these simulation models are very important. Other considerations is the information exchange. Unfortunately, in order to preserve privacy, the messages that are being exchanged between phones have to be quantized. Otherwise, if I sent a very, very precise real value, and then the other person colluding with someone else could potentially figure out that was coming from me, because it would be like a unique key that identifies me. Whereas otherwise, the only information a phone would have is, 6 days ago, I met this person for 15 minutes, and their risk level was 0.7. I mean, level 7, on 1 to 10, and that kind of quantization makes it impossible to trace back the information to me. Sorry, I didn't keep track of time. How much time do I have left?
Only a few minutes. You have to speed up.
Okay, sorry. So now I'm going to just quickly say a few words about this. This is my last slide about drug discovery projects. Just to give you a gist of what we're doing. So we're building neural nets that can predict the binding energy of candidate drugs or pairs of drugs with target proteins that the virus needs to be active. We are pre-training those neural nets by generating data from a physical docking engine that we have optimized to be very, very fast. So right now we can generate, for example, 200 million examples, which associate a particular molecule that's, say, initially randomly picked with the thing we want to predict. How well does the molecule bind to the target protein? Now, these are not the real binding energies, but they're pretty good estimators that can be used to pre-train networks. And then the molecules that look good then will be subjected to biological assays. There's also assays that are coming on currently. So in particular, Bill Gates paid Scripps to screen 20,000 existing molecules. we're working on screening pairs of molecules. So it's never going to be able, we're never going to be able to screen the 400 million possible pairs. So we have to use virtual screening to select out of the 400 million possible pairs of drugs, those that may be promising before they can be tested with biology. On the front of synthesizing new molecules, we're using an RL framework which we have been working on for a few months. That's an extension of AlphaZero, so it uses multicolored tree search, and it uses a representation of molecules based on rigid fragments. You can think of just the building blocks that form these drugs. And that makes the RL work really a lot better because you have sort of more abstract actions, and it also makes those molecules more synthesizable. And finally, one of the challenges in this thing is the true reward function would be like the biological assay, but that's too expensive. And so, and a slightly better reward function is calling the physical docking, but that's also very expensive, but not as much. It might be taking minutes to do that. So then we're going to train these neural nets that estimate what the docking would do. And then during the reinforcement learning, we want to have actions that say, I want to call this oracle, I'm going to pay a big price now to have an actual docking simulation rather than use the neural net, or even a bigger price to actually ask our friends that are working nearby to test that molecule for real in biology. So all kinds of interesting machine learning questions are raised. And this is just starting for COVID, but we have been working on this for other viruses before, so we are in a good shape. Thanks.
Thank you. Please stay tuned. We have a couple of questions. I have to also, Günther, perhaps if you go online again.
Yes.
I start with Joscha. Our question is blockchain a good tool that can help address some privacy issues for the app that has been developed by Mila?
It's a good question. I don't know. I don't have enough knowledge of blockchain, and I would need other people who know better to chip in. But yeah, I know that some people are exploring this path, and we're talking to a company that is building an app based on blockchain, but I don't know enough about the technology.
Joshua, another question for you. How would you handle false positives in the contact tracing app?
Well, that's the advantage of using machine learning, right? So we can make the true infection status a latent variable. We can use what is known medical tests to relate that latent variable to the observation of the test result, and then we can do the inference, using this noisy observation. So it's not a problem, and it will be reflected in the final risk estimation that you get at the end, along with all the other uncertainties that come in due to chances of transmission and symptoms and all these things.
Günther, a question for you. Are we working towards a cure for COVID or for vaccine for COVID?
Yeah, so I think the scientific community should do both. In my group, we're working on a drug, like a small molecule, but we're also doing work on antibodies. So I think a lot of people in the world at pharmaceutical companies are also working at designing antibodies against corona. So, and I think also both should be pursued. Both make sense. The cool thing about the antibodies is that you can now, so hospitals and so on are collecting blood samples, and people that are now immune because they have already had the disease, they have some information in their immune repertoire that we can extract and then use to either design a vaccine or an antibody directly. So that's a great advantage in the antibodies. On the other hand, in the small molecule world, the advantage is there's a lot of data around, and and a lot of methods already established in this area.
So yeah. I want to emphasize why it is important to do all these things in parallel. So the vaccines, we probably cannot deploy before a year because they have to be thoroughly tested. You don't want to give 99% of the population a drug that will kill them 6 months later. And so similarly, people are doing now single drug experiments because we kind of know very well what the dangers are with drugs that we have already tested. So if we find an existing drug, that's like the first short-term thing we can do. Then pairs of drugs, well, we don't need as much evidence before we release them compared to new drugs, which would be the next stage, and then finally vaccines. So there's time scales, and we have to do all the time scales because we're not sure which is going to work, and ideally the short-term things are going to work, but we're not sure.
Agreed. Yeah.
Günther, another question for you: is it possible to blend models that use different representation as input of a drug in order to boost the prediction?
Yes, sure. So I showed this a bit in a simplified fashion. So also one of the networks that I showed, this Deep Tox network that did the toxicity prediction, actually was an ensemble of neural networks and even other ML techniques trained on different representations. So yes, of course, with ensembles, you typically get better, and also some representations have the advantage and the disadvantage. For example, if you're in the low data regime, meaning you have maybe only a handful of each molecules of each class, then a deep learning approaches like learning a representation from smiles is hard. So there it might make sense to go back to the established molecular descriptors that already are known to have some relation often with drug discovery, or have some chemical knowledge already in there. The more data you have, the more you would go to the deep learning approaches where you learn representations. And in the end, of course, to make an ensemble of different approaches, like you say, blending, would give you an increased performance, and even small increases in performance could make a huge difference, because you can typically, after virtual screening, you have a rank of molecules, and you can typically only screen or test a really small fraction of those. And if your predictive performance is only a bit better, then you have a better chance to get a good molecule in your top ranked list. So yes, exploit everything to get good models.
Joshua, a question here. Are you aware, or are you in contact with the Trace Together efforts in Singapore?
Yes, we're obviously aware. I think they have the credit that they were the first ones to do something where they tried to get some privacy using a peer-to-peer approach with Bluetooth. And we're trying to get their code, because they haven't really released their code, even though they promised to do it. We might incorporate some of it for the Bluetooth aspect. So yeah, very important. And there's a lot of collaboration around the world with people developing these things, which is pretty amazing.
Cool to hear that. Another question for you: have you considered co-localization to detect when pairs of phones are close together?
Well, that's what we're trying to do. But the question is the technological question. If you use things like GPS, Wi-Fi, and cell towers, the precision is not very good. It's not good enough. It's like maybe 10 meters. I mean, it depends where you are in a city and so on. Whereas if you use Bluetooth, this is very precise. Another example to see why Bluetooth is much better, if we can get it to work, is think about a big building like apartments. People are all in the same XY location, and they would register as contacts, but obviously they're not. But if Bluetooth, if you're within 2 meters, you can in principle figure it out.
Perhaps a question to both of you. Which exact proteins, I think Günther already told it, of the virus do you target during your drug discovery effort?
Yes, I would love to target all of them in this short approach that we showed. So it's a very restricted study. There was data on two proteins called proteases. These are proteins that are very vital at the beginning of the replication cycle of the virus. So when the virus enters the cell, it's an RNA virus, and then the human ribosomes already start transcribing this RNA, the viral RNA, into a viral protein. And you have a polypeptide chain that doesn't work, but there are two proteases. They're called 3CL-like and PLP protease, and they start working first and cleave up the polypeptide chains such that the other proteins, or that the machinery of the virus starts to work. So they're relatively early in the replication cycle. And that's, I think, why these assays were designed to find inhibitors of those two proteins called proteases. But of course, there could be also, like you could also target the structural proteins or the RNA polymerase. So there's quite a couple of molecules, even if it's simple and there's only a handful of molecules, all of them can be targeted. And so in our approach, the consensus score should find molecules that inhibit both proteases. So you hit them, or you already try to hit the virus at two positions, but yeah, so all approaches are possible here.
Joshua, you want to add something?
Yeah, we're also working on a protease.
Yeah, great.
I could, but Joshua, I have specific something for you. In the future, how can we detect something like COVID-19 kind of viruses in the very early stage using AI or using machine learning? And how can we then prevent an outbreak, or how can we help in the very early stages?
Well, I think the tracing is aiming at that, right? Because even the problem is we want to be able to identify risky people even before they have symptoms. And so if we keep track of your encounters, that's a very strong clue. But there may be other measurements. So there's, before you have serious symptoms, you might have something changing in the way your breathing is going on because it's happening in your lungs. And there's a project by Kyung-Young Cho, who's a faculty at NYU and one of my former postdocs, who they're training machine learning models to predict that you're infected based on the sounds of your breathing. And this seems to work, but they're still like in data's early stages.
Along this direction, there's also a question for you, Joshua. What do you think about deep learning-based approaches in terms of imaging, CT imaging, X-ray imaging for diagnosis?
Yeah, there's a lot of effort going on. It's one of the things I've discussed in the review paper that I mentioned. I mean, the issue is, you need people to go through the CT scan and so on. Ideally, what I would like is something, for example, that would work in poor countries where they don't have these equipments, and just using the phone, using maybe the camera and the audio, can we get some clues that you might be infected? Of course, with the contacts as well. But even the phones is a problem because in many countries, only a small fraction of the population have phones. So it's so one of the things that I think can be done is also provide services by telephone. So it doesn't have to be your personal phone. You can call a number and then interact with a website, which maybe using dialogue, asks you questions and give you recommendations, nudging, and so on.
For the sake of time, I have to switch on. Thanks a lot to you two guys for your contribution. It was great. And I hand over to the next chair.
Thanks, Sepp.
So thanks a lot. So let's go on to the next session. So I'm happy to introduce the next speaker. It's in the session of healthcare management. COVID puts a lot of strain on the healthcare system, and there are many opportunities how machine learning could be used to help doing that management more optimally. So our next talk is going to be given by Michaela van der Schaar. She's a professor of machine learning, artificial intelligence medicine at the University of Cambridge and a fellow of the Alan Turing Institute in London, where she also leads the data science efforts there. So Michaela is very active in developing machine learning models and applications for medical data. She's also helping to build the ELLIS community, and together with Oliver Stegler and me, she's co-directing the ELLIS Health program. So today she gives an overview talk about how artificial intelligence and machine learning can help the healthcare system to respond to COVID-19. So Michaela, I'm very glad you could make it, and I'm looking forward to your talk.
Thank you very much, Gunnar. Hopefully the slides are visible now.
Yes, they are.
So thank you very much for giving me the opportunity now from the discussions about prediction to the development of technologies to help hospitals cope with the current crisis and build tools to enable clinicians and hospital managers to deal with this. This work is a collaboration between my own research lab, Machine Learning and Artificial Intelligence for Medicine, where Dr. Ahmed Allah and Zhao Zhiqian are working with me to develop these methodologies. We have a complex team where we have Ari Ercole, quizzes, giving us input as to what are the useful technologies for healthcare management, biostatistics, and this is a collaboration with Public Health England and NHS Improvement, because we hope that these technologies are going to be scaled at the level of the entire NHS and hopefully beyond. So our goal in this project is to provide evidence that can reliably assist the difficult decision-making process that clinicians and managers need to face nowadays to save lives. The focus here is not to let machines make decisions. The focus is to use the available data to provide the right evidence to clinicians and managers such that they are informed about how this disease is best managed and how it's evolving. So as part of that, what we are doing actually is using depersonalized Public Health England chest data that I'm going to briefly present in a little bit, and for decision-making risk for each patient who already has been identified with COVID, identify what is the best treatment plan for these patients, and also what is the relative benefit from using a particular type of resources and when. This is also useful for resource planning at the local level in the hospital, such that individual hospitals can identify what type of resources may be needed and when. So the type of decisions I'm talking about are, upon admission to the hospital, COVID patients may face different type of events. And healthcare professionals need to make decisions as to when to transfer a particular patient to an intensive care unit, when a patient should be put on a ventilator and what type of ventilator, potentially for how long, when patients can be discharged, and when patients can go safely home. Because the resources are increasingly limited, both in terms of ICU beds and ventilators, and as you will see a little bit later, it seems that it is important that the patients are rapidly put on ventilator and admitted to the ICU. This is increasingly becoming an important issue. We also would like to understand what resources are needed, and we would like to do that not only at the aggregate level like some colleagues are doing, but rather at the personalized level and at the hospital level, such that the current demand and the forecast of subsequent demand can be in a very granular and accurate way modeled. For this, we are collaborating with Public Health England, which has commissioned a very important and very powerful data set called CHESS. CHESS stands for COVID-19 Hospitalization in England Surveillance System, and contains information of patients that are already hospitalized through the UK Public Health England system. So this is data that's across various regions in the UK. And the data that I'm going to present you results on today is data collected until March 29th. The information that we have contains information about personal information, some laboratory details, some hospitalization details, risk factors such as comorbidities, and the outcomes we have so far. Note, though, that this data, of course, is heavily censored because patients are still in the hospital. So in terms of demographics, the type of patients admitted tend to be older, and this is unsurprisingly so. It is interesting to to see that admitted to be predominantly and again this it's also interesting to see that at least for now in the UK from other hospitals. So for now, at least, it seems that the capacity is sufficiently there, such that patients are directly admitted to the hospital rather than being sent from another hospital. In terms of statistics, again, the number of patients we currently have that are COVID patients in the hospitals are around 1,700, and out of this, 650 are admitted already to the ICU. And a percentage of them is on ventilators. It is also interesting to see that a large majority of patients that are admitted are, of course, still hospitalized. It is what you are seeing on the left graph is that most immediately to ICU after hospitalization is very high. So many patients are admitted to the ICU almost immediately. So who is being admitted to the ICU? You have some distribution across age, gender, but also different comorbidities. It is interesting to see that the patients that are not admitted to the ICU tend to be a little bit older. In discussion with our team, part of the reason for this may be that some of these older patients may have statements that they do not want to be admitted to the ICU or they may not want to be resuscitated. But there may be also some inherent bias where some of the older patients are not given the same resources. Who is being ventilated? Again, you will see a variety of metrics here and across comorbidities. What are we doing on the basis of this data? We are using a set of machine learning methods that we have developed over the years that are automated machine learning frameworks that encompass a variety of machine learning methods that have been developed by a variety of groups over the years. So these automated machine learning methods are creating ensembles of many, many machine learning methods developed so far, as well as data imputation methods. And what we do here is we do both a multi-class prediction as well as a time to event analysis. So what we are predicting is whether a person would be, what is the risk of a patient to die, to be discharged, or to be admitted to the ICU, or be put on a particular ventilator. And we are also looking at the time to event, time till this particular event, as well as the time that a particular patient is on a particular device. So as I mentioned before, what we are forecasting is the personalized risk for each patient of death. But also risk different types of events, as well as when patients may be admitted to the ICU and ventilated, are identifying causal inference methods that we have developed in our group, benefits of treatment. Definitions about the current analysis and what the current methodologies in machine learning can provide in terms of information for their own understanding. Because we are predicting not only when patients will be admitted to the ICU, but how long they will stay there, as well as how long they will be on ventilators, this is also very useful for local hospitals to do future resource planning and know whether it will be possible for them to admit a particular patient, or whether this patient, given its predicted risk of needing a particular set of resources in the short run that may not be available in the current hospital, should be routed to another hospital which may have these resources. So at every moment in time, we can say on the basis of these machine learning models, what is the current utilization of resources, scarce resources in the short run, in the long run, and this can inform resource planning, but also admission to the hospital or rerouting of these patients. How well can we predict so far? So it seems that our model, which is based on a machine learning model, as I was mentioning before, based on a technology called autoprognosis, is using all the features, the prediction mortality within 1 week, is that if we look only at age, the predictions drop quite significantly. And if you look only at age and number of comorbidities, the performance is also somewhat lower. So what seems to be important is to look at age-specific comorbidities, but also the time from the symptoms to hospitalization. Again, two other points to note, as opposed to conventional statistical models that are linear, like Cox regression, the performance improvements due to machine learning seem to be quite significant. And this is unsurprisingly due to the nonlinear interactions that are clearly appearing here. Also, if you are comparing with Charleston index, which is an index that has been used in some countries to deny access to service, you can see that this outdated comorbidity index is really not useful at all in this context. If you are interested to see what variables have contributed to these predictions, you can see that age is indeed predominantly important and different morbidities play an important role. Now, if we move more towards predictions of ICU admissions, you can see that we have considering multiple features and looking at then prediction of ventilation. We are predicting ventilation, but also what type of ventilator will be used. And again, you can see that the predictions tend to be really quite good. So on the basis of this, we built a demonstrator, a very cautious demonstrator, for the sake of clinicians to be able to input information about their patients, such as, for instance, gender, and be able to identify what is the prediction of mortality in the short run and in the long run, but also prediction of discharge, as well as confidence associated with it. As the patients may have different types of comorbidities as well as treatments, these predictions can be adapted. Finally, and importantly, probably the biggest use of this particular type of technology is also on resource planning at the hospital level. So what you see here is the past admissions up to March 25th of patients in the hospital. And what we are able to issue at the hospital level is how these patients that are already admitted in the hospital, what will be the resource utilization in terms of both beds in regular wards, ICU beds, and ventilators, and what will they require in the short run and in the long run. So finally, machine learning. One of them is only measurements at admission to the hospital. And even those measurements tend to be coarse. They contain comorbidities, they contain the time till symptoms, they contain some demographic information, but not lab tests in the sense of blood tests. Also, we do not have measurements after they have been added to the hospital. We just have events that have happened, whether the person was discharged or the person died, when they were admitted to the ICU, when they were admitted to a particular ventilation device. So as a next step, we hope that such information may become available. That may not be available necessarily in a particular country. So it's clear that we need to increasingly join forces with others to analyze such data to better help clinicians. Also, in the past, in our past research, a very important issue has been issuing early warning systems. in our extensive discussion with ICU doctors here in the UK, it appears that some coronavirus patients are unexpectedly and in an unexpected way deteriorating and doing badly. So we need to understand why these patients are unexpectedly deteriorating and provide that information to clinicians such that they can keep an eye on these patients and provide the necessary resources. Finally, we are increasingly looking at treatment effects and causal inference for treatment effects. So what is the effect of giving a ventilator earlier or later? This seems to make quite a difference. We also hope that through ideas on inverse reinforcement learning, we can inform policies and improve collaboration among clinicians. We also need to manage uncertainty because this disease is changing. The way in which they are treated is changing as well. So we really need to have methods such as the automated machine learning methods that I was describing to be able to update these predictions continuously and understand when we have shifts in prediction. And also, last but not least, there are other ways in which I believe machine learning can help, inclusively identifying what type of treatments, new treatments should be given to patients. And I posted here under a set of challenges that our Cambridge Center for AI in Medicine has identified as being vitally important in the short run. So thank you, Gunnar.
Yeah, thanks a lot, Michaela. Thanks for the great overview. Okay, so we have quite a few questions online. So let's start with the first one. Apologies to the audience. We had some issues with the audio. I guess we're all competing with our teenage kids to download videos and software and so on. Okay, so there's one question about the data availability. So are parts of the CHESS data set publicly available? For instance, the ICU COVID usage summary statistics per day and so on.
So unfortunately, while this data is de-anonymized, it is not publicly available. I find this personally very unfortunate. Currently, that is not the case. If that will change, and I truly hope that it will change, I can inform the ELLIS community and the more general community about it. Currently, that is available only to academics and only some academics within the UK community.
Is that a legal constraint, or is that just the ethics is missing?
To be honest, I do not completely understand why it is not made more widely available.
Mm-hmm. Okay. If there's any questions from the audience in the room, in the Zoom meeting, just go ahead, ask. Otherwise, I go on with the next question online.
Michaela, is the demonstrator accessible publicly?
Yes, we are working, not yet. Thank you so much for this question. We are working very hard to make this available. We need to be very cautious because we do not want that this tool is wrongly believed to make decisions about who should get ventilators, who should get to the ICU. We are just providing evidence to our clinical collaborators as to what are the findings so far and providing this information to them. So we are working very heavily with both ICU collaborators, clinical collaborators, as well as with experts in healthcare management to understand what's the best way to provide this information. And there may be information for both clinicians, but also information for hospital managers, because we would like to be able for them to have an understanding of local individualized predictions of capacity. So many very good models exist that do capacity planning at the global scale, at the national level. Our view is a micro view where we are making these predictions on the basis of the patients currently admitted and doing that on a patient-by-patient basis. And this is how we create these local hospital aggregates. So hope is also that we will provide a demonstrator for that, which hopefully can be linked with the demand management methods that have been proposed by others at the population level.
Okay, here's another question from the audience. Can you talk about the... Oh, go ahead.
Yeah, if it's okay. Oh, sorry.
Sure.
Yeah. So you were talking about not misusing the index. I think this is really great. But there's the other index. I think it was like the Charleston index that that was performing very poorly, but was still you had evidence that it was being used. Are you making a really strong statement against that? A really strong and public statement to avoid its use?
Yes, I'm making a strong and public statement against its use. I think the analysis, there have been many studies even before COVID where Charleston index seems to be an outdated index that was developed many years back on the basis of very different evidence. And just looking at number of comorbidities without looking at the type of comorbidities, their interactions, when these comorbidities possibly have occurred in a patient, I think that this is a mistake to make rationing decisions on the basis of an outdated index such as Charleston, yes.
Okay, I have an online question from Julia. Can you talk about the big difference in gender seen in the data? Where does the big difference come from, and are there less deaths among the women?
Oh yeah, that is a very interesting question. And this is something that I really hope to have more data about soon. This is very interesting. So it is clear that less women are admitted in the UK in the hospital. This may be both because women may experience less severe forms of the disease, but there may also be possibly bias associated with it, meaning the women may not be as strong advocates of themselves to request admission to the hospital. So they may be, this may be also some type of bias where women are not demanding this admission. We do not know that. What is interesting is that it seems that women are not protected with respect to dying. So in terms of deaths, the percentage is similar in males and females. We are clearly paying attention to this gender bias because it's interesting. What seems to be the bigger predictor of mortality and of severity of disease are some of the specific comorbidities involved, whether they are related to asthma, whether they are related to hypertension or severe obesity.
Maybe can I follow up here? You also had a surprising peak for young children in hospital admissions. Do you think that's the same effect that maybe the parents are more likely to bring them to a hospital?
No, so I do not know really about that. This is something that we do not know right now, whether potentially there was a coding error associated with it. So as a matter of fact, we have excluded in some of our analyses the very young children. So that's a good point. We do not know there whether we have a data quality issue or not.
There's actually many more questions online. So maybe I just pick one here. So how do you know that the model does not prefer resources over lives? So maybe that goes in the same direction. Maybe you can address this general topic.
Yeah, so the focus here is not, the whole purpose of this tool is not to ration resources. The whole purpose of this tool is to try to understand better what's the current evidence about the disease trajectory and understand what resources are needed and the value of these resources, not with the idea of rationing them, but with the idea of forecasting very accurately local demand and hence requiring additional demand, additional resources for patients that may need it. So again, the focus is here more for information, planning, and understanding of the disease rather than rationing resources.
Yeah. Okay, I think we are over time now, so I think we need to move on to the next session. Michaela, thanks so much. There's many more questions where you can have a look at them later.
Thank you.
Thanks.
Great. So the next session is on data resources and challenge community platform. This is really going back to what we stated at the beginning of this workshop to also provide a forum for others to engage in understanding what the resources are. There were many questions about data sets and opportunities, and some were already answered in the presentations. But we have now two brief talks on more specific initiatives that have been launched in different ways. So the first speaker is Freit Hambrecht, who is going to present on an initiative that he spearheaded and put together, which also touches and interacts now with others to basically establish a platform where individuals can bring questions or expertise or data, and which aims to bring this all together. So yeah, without further ado, thank you, Fred. Over to you.
Thanks, Oliver. I'm speaking on behalf of a bigger crowd of people who all had the same impulse, the same basic idea, namely, as you see in our logo, we have to tackle this disease, this pandemic on different timescales. In the very short term, there is the allocation of vital resources, of limited resources. There's the question of optimal treatment strategies in intensive care units. We need to make good epidemiological predictions to inform policymakers. We, on the longer time scale, as we heard, are interested in developing drugs and vaccines. And luckily, us as machine learners, in a broad sense, we will be needed, or we are able to make contribution at all of these stages. And in talking to people from our community, to you colleagues, there was a unanimous sense of really wanting to contribute, of wanting to help. And well, we thought one way to bring us together with people who are working at the front, medical doctors, biologists, and so on, would be to create a free exchange where questions and people who may be able to give answers, so they can come together. Now, some of the issues that we've heard about here and that I've briefly listed, they entail questions in operations research, in bioinformatics, in machine learning, computer vision, GI science, cheminformatics, statistics, and so on. And luckily, we have found volunteers from all of these areas. So there is a list of PIs, so real people who tend to be much better at coding and doing the real thing than the PIs. They have volunteered to offer their expertise to whatever questions may be thrown at them. And well, we want to make it very simple for people to come to us with questions. Yeah, so we mainly here cater for these folks, people who have COVID-related data and or questions. And there are two ways they can get in touch and ask for help. The simplest one maybe is by filling in a very simple form where you tell us what kind of help do you need and how quickly. Alternatively, you can also directly go to a discourse that we're hosting. This discourse is organized in a number of categories, namely, I have data and or analysis problems, questions, I have issues with my software stack. People on our platform may be able to help, but there's also a platform that has sprung up to concentrate on just that. And on the other hand, there are the people wanting to help. And we luckily so far a number of things that have worked. So a few problems that we could solve already. And if I can find my tab hidden under the Zoom controls. So we have a few issues that we can solve as, that have a status of being solved, where askers and experts came together and could solve a problem. We have also, I've seen in the chat questions about, list of resources. There are many of these on the internet now, and we all believe it's good to have a little bit of redundancy. But we have such a wiki also where we try to give a fine-grained categorization of data sets, services, dashboards, and you can browse this, and you are free to make pull requests or suggestions on how to improve that if there is something. Of course, this is developing very, very quickly. It's everything on a scale of hours and days here. This is, I think we are reasonably set up now. This will scale well with the number of requests because everything is, as an open source development, everything here is transparent. People from around the world can work together. But machine learning scientists, to do their best well, they need data. And as we heard in some of the talks, some of this data has real privacy issues. Sometimes there is just an issue of the people who have the data being completely overworked and not being able, not having the time, resources, the energy left to pass it on to others. And this is, I think, a real limiting resource of the amount of data that we as a community have access to. So what can you do? Well, if you want to have news from this platform, you can follow this on Twitter. You can register as a volunteer yourself by, visiting our landing page and making a post here in a category where many people with great expertise have already presented themselves with what they think they can contribute. The most important thing, though, will be the following. By this 6 degrees of separation idea, probably you know someone or you know someone who actually has data and problems. And sometimes you will be the right person to deal with part of those questions. So maybe you're a computer vision scientist, maybe you will know exactly what to do with the chest images, but there may be other data associated. For example, there may be other biomarkers that were recorded along with the chest x-rays, and you may not be the right person to address those questions. So whenever there are these whenever there's this extra data or extra information, then tell people or be yourself the one to bring these questions to the platform to allow others to do their share in helping fight this pandemic, which is unfortunately threatening so many friends and family of us. Thanks, Oliver.
Thank you very much, Fred, for this very short talk. Again, we will talk questions at the end and have maybe also span out a more general discussion about platforms and data sets. So I think I would now hand over to Deborah Ritchie, who's going to join us today from Kaggle initiative, and will present some of the existing opportunities to engage here as a platform for addressing analysis and also challenges related to COVID. Thank you.
Yeah, excellent. Thank you very much, Oliver. And wanted to present my screen. I'm just running into an error about starting screen share while another participant is sharing.
I will stop sharing, and there you go.
There we go. Thank you very much. Thank you, Oliver. So as you'd mentioned, I'll be presenting on behalf of Kaggle. So Kaggle, for those that aren't familiar, is a machine learning platform and a community of about 4 million data scientists who come together to solve tasks about real-world data science challenges. We've hosted a number of initiatives recently, specifically around better understanding COVID-19. And so if you were to come to our landing page, I think you'd notice off the bat a couple of different items. The website itself is a platform that contains the different tools that a data scientist might need to begin a project. So we both have access to a large number of public data sets, both related to COVID-19 and completely adjacent, as well as a web-hosted notebook IDE environment where users are able to run code and train models based on free compute resources that we offer, either on CPUs, GPUs, or TPUs. We have a special initiative around COVID-19, and I wanted to walk a little bit through some of the projects that we're doing today and some of the results that we're also finding in our early work. So if everyone can see my screen, you'll be able to see kind of our landing page that we've set up specifically for COVID-19 and 2 key challenges that I think might be relevant for the data science audience. The first is a challenge that we've set up on behalf of the US White House, as well as a number of other organizations like the Allen Institute for AI, Chan Zuckerberg Initiative, Microsoft Research, Georgetown University, and Google Cloud, and that's to take part in a COVID-19 NLP challenge. In this case, there's a data set of over 45,000 medical research articles, and the goal is to be able to essentially apply text mining techniques to understand a range of actually 70 different questions that have been given to us by the WHO and the National Academy of Sciences in the United States about COVID-19. This challenge has been a key part of really a lot of our COVID-19 outreach, and has actually gotten a large amount of responses so far. In just a minute, I'll walk you through what some of the early results on that challenge have looked like. The second thing that we've also done is launch a series of weekly COVID-19 forecasting challenges. This is to both forecast the potential total number of cases on a regional basis, as well as the total number of fatalities. This challenge has been running on an ongoing weekly basis, starting a week and a half ago. And we expect to have the first set of results in terms of forecastings within the next 2.5 weeks. Lastly, we have a large number of COVID-19 related data sets overall on the platform. And so we always encourage users to be able to upload additional data sets that they might find useful or that they think that the community might find useful. I wanted to briefly walk through what some of the contributions so far from the Kaggle community have looked like regarding COVID-19. And so if I can move my Zoom bar. So the COVID-19 Kaggle community contributions have really looked in 3 different dimensions. The first is what users have been able to find within the literature. And so I mentioned that the first challenge was really around text mining to be able to extract out insights about COVID-19 from a large body of academic publishing. And so if we look at aspects that the WHO have told us that they're interested in, for example, information on transmission, incubation, environmental stability, you can see what a number of results that we've been able to pull out, our Kaggle community has been able to pull out essentially about, for example, the incubation period, along with the citations of which papers they may have come from, along with any relevant confidence intervals, factors like the prevalence of asymptomatic shedding, and what the different insights here are. And generally speaking, we've collected like a large amount of data that we've been told from medical professionals will be helpful for us to be able to extract from the articles. And so you can see this continues with risk factors. Another set of things that we're also highlighting as contributions are search engines. So these are question answering engines or search engines over the body of literature that allow users to explore COVID-19 related data in a more systematic way. And then finally, as we've been talking about before, access to data sets. So we have access to a large amount of different public data, including regional and city level data from Johns Hopkins University, but also a number of individual data sets that might be related to COVID-19. So for example, the number of ICU beds broken up by county, or the number of COVID-19 tests. All of this is community provided. It's uploaded by different members of the Kaggle community so far. And we've been really excited by some of the early work that's started off here. If you're interested in contributing, as soon as you make it over to the Kaggle homepage, you'll be able to see some of the early steps in terms of how to get started. Thank you very much.
Thank you. So this is the last session that's coming to an end. I'm just wondering whether there are questions in this area. I'm first maybe handing the opportunity around to people here on the Zoom channel. If there are questions or comments that I'm seeing on the Google form, if you have questions, also please type in your questions now, or also comments for other activities or how to collect them, anything that might be useful is there. But first of all, here, is there anybody who would like to comment on this?
I mean, I think these resources are really great, and I think we should continue collecting those, so people, because this is where people can really get involved and get active to do something. So I love the Kaggle initiative. I think there's a few other ones which you should give it the opportunity in the future also to present their stuff. So I think it's very good to have data sets together with questions, and I think I really like this how it's done on Kaggle. And we need more of that. I think we should look for the next presentations in the next iterations of this meeting to have more of that.
Maybe a question that's related to this. I mean, we highlighted this before, and I think also several of the speakers made that remark, that a challenge at the moment is data. And we could also maybe maybe also other speakers can comment on that, where we expect that the larger data sets are going to emerge. I mean, some questions will remain difficult to share specific private data. But in other areas, the problems will just sort out themselves, and data will come our way. And if there is sort of maybe you already have a sense of where future data that are coming or others, because I think that would be quite an interesting perspective for those who are thinking about whether there are specific questions or challenges to solve.
You are asking me, Oli?
Yeah, that's great. I mean, the speakers.
I think that definitely we need to try to understand. I think, like Joshua Penjo rightly said, there is a trade-off here between privacy and the value of the data. Definitely, as long as some of this data is well protected and we cannot identify who the patients are, then hopefully some of this data can be released. But in many settings that contain healthcare information, it is quite easy to identify possibly from the time somebody was admitted or time somebody dies who the person was. So I think that what's important in this regard is, again, machine learning community has started to look at ways of generating synthetic data which have good fidelity, yet have differential privacy guarantees. So possibly one way to think about this in the future would be to look at innovative ways using machine learning to generate synthetic data of high fidelity, but which have privacy guarantees, strong privacy guarantees. But I would be eager to hear what others have to say.
Speaking quickly from Kaggle's perspective, I think we've actually been, we would love to see and we want to see more organizations continue to host their data related to COVID-19 on the platform. But we've actually been relatively happy in terms of the influx of new data sets that we've seen related to the pandemic that exist. I think one of the things that some of the government organizations have pointed out is just getting more eyeballs onto some of the existing data sets will be really helpful. So for example, in December of this last year, there were more than 2,000 research articles published related to either COVID-19 or coronaviruses more broadly. And of course, it's difficult to be able to go through all of these articles in order to find answers to different questions that people in the global health organizations might have. And that's kind of in a large part what motivated one of our challenges, which was there's a large body of publication, large body of data that we haven't had an opportunity to look at systematically. And we want to open this up to the data science community to help us digest.
Maybe another question, synthesizing here from the comments we received online, but also here from the chat window, which Ernie Lawrence pointed out the question, differences between developed and non-developing developing world. I mean, what data sources are available and how could this also help to engage with the developing world where these resources, this beautifully enclosed NHS data resource we heard from Michaela and others, this just doesn't exist there. Is there a role for that? Maybe linked to that, there's also an online question which asked about data sets from Asian communities. I assume that is represented, but maybe you could also comment in general, what's the representation of data from which nationalities, countries at the moment, and maybe then the link out to the developing world, if you have anything, or same for Fred. I mean, maybe you could both, see what's already available on your side as well, also for these platforms, what's the diversity representation at the moment? That's maybe also a good question.
Yeah, I can start maybe with a quick response to that. Generally speaking, I think on Kaggle's platform, we could use more data from the developing world. I think the fidelity of the data that we have that's highest today is really, especially from the United States and some of the global data that we have from Johns Hopkins University. In terms of what types of data we do have though, from other countries, we have, for example, data sets on like ICU beds and hospitals numbers in India, for example. This type of data ends up being relatively popular. Generally speaking, we're very reliant on our community to upload data sets though. And so our data does bias in the direction of our community's preference.
Yes, so we have heard from Next Strain that the sequencing is starting to come in and scale up, and this is great. But there are so many other heterogeneous sources of information, and this is why, by the way, we created such an, if you will, unstructured platform that would accommodate all kinds of heterogeneous data and questions. And overall, I think there is still a dearth of data. So I've tried to reach out through personal contacts to China, and the informal answer I got was that there seems to be hesitation to share the data outside the country. I cannot comment on if this is true or what's the underlying cause. Certainly, yes, if there's sequences not just of the virus but of the humans involved, there are huge privacy issues that we need to consider. But for other kind of data, frankly, I don't understand the problem. So we're talking, for example, we're talking chest images here. We know that face images can be difficult to anonymize. So if you're working on the head, this is one thing, but with chest, it's quite another. And maybe it's naivety, or more likely it is the data being stuck somewhere where people are overworked and too much, or they have to focus so much on the everyday problem that they cannot find the time, the energy to put it out for others to help. So this is why I invoked this 6 degrees of separation. If you don't have data, but you may know someone who has, then you could be the person who gets the data on a stick or whatever, and if legally possible, post it. Or if you cannot post the data, at least you can post the question, and we can find in secure channels. We can connect you to people who are knowledgeable in that kind of data, and will hopefully be able to help you.
There's another question here related to this, which comments on heterogeneity in gathering and registering data, a number of infections, tested, deaths, and so forth in different countries. I think that's a general question. Probably that's not really related, frankly, to your platform, but in general, which points towards the heterogeneity of quality and also different measures and methods to ensuring comparability. I mean, to me, it sounds almost more like a scientific question, how to bring these data together. But anyone who has a thought about that, or whether you're tackling these data sets, would be very interesting. Also, we know that the testing rates differ greatly across countries. We've heard about that, and obviously that makes it somewhat challenging to interpret these data.
It is also, if I may comment, Oliver, it is not only heterogeneity of data, but also the fact that the disease is changing to some extent, and possibly in terms at least of healthcare systems, not only the utilization is changing, but also because the disease is changing, the way doctors and managers of hospitals react to these resources are also changing. So I think we need to be understanding also drift, even in the current data we have. But in terms of linking data sets, I think that's an enormous opportunity to cross-check ourselves. There are at times opportunities to link diverse data sets, not only to make predictions better, but also to validate our findings and cross-validate our findings, as well as the accuracy of these findings and of the data we fed in. So I'd really like to encourage the community to think deeply about how to deal with drifts in data as the disease is changing, to identify those, because that I think it's important, but also to link these different data sets to check and understand whether we are doing something correctly or not, as well as check the quality of the data that we have.
Yeah, these are good comments. I mean, maybe one thing to add here. I mean, I think we haven't managed for this conference to get someone from this representative groups, but there are also efforts on the way to try at least within Europe. Obviously, there are different efforts in different geographic areas, but within Europe to harmonize data across country boundaries. There's a larger effort launched and supported by the European Commission, linked to the European Open Science Cloud, where ELLIS here and EBI are in a driving role, to try and help there and create infrastructure. And I think there's a lot of will to do this quite quickly. So I think we will hopefully overcome some of these bottlenecks in sharing data. But at the moment, that seems still a major bottleneck, right? I mean, to make data available on a larger scale to a larger community under controlled security conditions and other elements that we can tap into that. I've just seen, Neil, you put a number of comments relating to Africa and cases there. Do you want to, tell more about this if you know something about this, or is it just just sharing these links? And it looks quite interesting. So
yeah, it's something that Data Science Africa, we've been looking at putting together a virtual conference, I think for this week or next week, should be similar to this, focused on the African context. I'd like to mention, like in particular, Elaine Nsoeisi, who's public health at Boston, who's been doing a lot of work looking at data collection efforts in the African context. Particular challenges are, even if we deal with the virus in Europe, if there are countries where the virus sustains and mutates, then even if we gain the infamous herd immunity, there's the possibility that the virus can come back. So it really is a world problem. And while I know we're very focused on efforts to contain within more developed contexts, if we don't give the support or encourage teams in the African context to be able to develop their own solutions, I think it's going to come back and bite us again.
Yeah. Are there any other general comments? Thoughts?
I had a question, maybe for Mihaela. This is Saj Mukherjee. Can you sort of say anything about in the PHE data? What fraction of hospitalized, ICU, ventilated, etc., have none of the pre-existing conditions? So I guess my question is really about sort of how, how effective is the simple binary rule that, one or more pre-existing conditions, age cutoff, yeah.
So this is a question to me, if I understand.
Yes.
How well does such a predict? Not very well. Not very well. So just by looking at age and number of comorbidities, the predictions are not very good. And definitely not the predictions of who will need ventilation, et cetera, but even the predictions of mortality, as we have shown, it is not so much only age is clearly a very important factor, but age alone, without looking at the type of comorbidities involved, is not sufficient.
Thank you.
Yeah, just maybe two more comments. There was one question directed to myself regarding interactions with an existing competence center for data quality, St. Gallen, and I think regarding whether the interactions plan, I think the interactions with data generators or initiatives to consolidate and curate data and make them available, that's an important element. And I think we hope also to get any comments on the web page so people are aware of activities that generate and consolidate data. It would be great to let us know, and we will put them there, and maybe they could also feed into these open data resources and Kaggle or in controlled access data resources and otherwise. And also thank you for those who sent comments to Fred. We will forward them all. So if you send us comments, you could we forward them or obviously contact Fred Hambrecht directly.
Maybe I can add, some people are sending comments and asking for a reply, and it seems like they don't include their addresses. So if I understand correctly, we don't automatically have the addresses unless you include them. Yeah.
I've started to respond to a couple of questions on the Google Sheet directly.
Yeah, there's in total there's 140 questions or something overall, so it's quite a large number. We cannot address all of them, so I think it's good if we can do this by email afterwards. So
yeah, we'll go through.
Really helpful. But in any case, we will share, I think, the questions and comments with the speakers, so they have the feedback directly, even if we couldn't address it here.
Yeah, I don't know. Maybe if there's no question right now, it's also a good point where we can just say a little bit about how we put this together. So this was a totally chaotic last-minute effort where we just asked around who knows somebody who's doing something interesting, and we're using these contacts to put together a program. But maybe if we do this again, and it would be interesting to hear how people feel about this, we can start becoming a little bit more structured, and we have more time to get suggestions of people who they think should be speaking in the next edition of this kind of event. Any feelings about that? So why don't we say whoever has an idea or a recommendation who should be talking, please send it to the email address that's listed on the ELLIS page. And also if you have an opinion about whether we should do it again, how often we should do it, please write that also. Either email it to Daniela, who will receive this email, or put it in the Google form. I think we also have to thank everybody who helped organizing this. Right, so this I don't know, maybe they can switch their videos on for a minute so we can see them and say hello. So the focus changes to their faces.
Hello.
And also the logistics team.
Hello.
Vincent.
Hello.
Arash, yeah, and
hello. Yeah. Hello.
Joanna and Daniela, of course. I don't know, Gunnar, Oli, Michaela, or sorry.
Maybe we should thank people from Zoom that this works so well.
It worked reasonably well. So I think unless the audio quality is not perfect sometimes, but you can't do that much. So I think we will work on how to maybe dial in via phone, so it could help a bit to improve the quality for some speakers.
Yeah. So I have
we had overall 1,300 people online, I think, on YouTube, and about 90 people in Zoom. So overall, this worked really well. So.
Yeah, it was amazing. I mean, we had this half hour at the end that we reserved for questions, anticipating that people will go over time as we scientists usually do. But it feels like there was this sense of urgency that everybody was only saying what's absolutely necessary. It reminded me of once. I remember when I was a PhD student, once I was in a session which was chaired by my PhD advisor, Vladimir Wapnik. And in that session, everybody finished their talk in about half the time. I guess they were sitting there intimidated by Wapnik in the first row and thought, I don't have much to say, and just rushing through their slides, and everything was finished after half of the time. So it feels a bit like it was the same. Everybody feels, I don't want to steal time from all those people working on these projects. So everybody stuck with their, more or less with their 15 minutes. And I was very impressed. I thought this was amazing display of the creativity and ingenuity and also motivation of the community that is trying to take on this problem. And I hope that many got new ideas from this and maybe new motivation what to do. I don't know. So anybody else wants to make any concluding comments?
Yeah, Ella, you wanted to say something at the end?
No, all I wanted to say was that I really hope that the community is eager to continue this type of discussion and collaboration. And I think that we, especially the ELLIS Health group, we would like to hear from you as to ideas how to make this more useful and more interactive. So we would like to receive comments as to how to improve what we have done so far and how we can create a forum that is truly useful for everyone, definitely for the Europeans, but of course for everybody, even outside Europe, and how we can better collaborate and we can use this crisis to build networks, to foster research that hopefully not only will conquer COVID, but will last beyond COVID. So we like to really welcome the community to send us feedback and ideas as to how to further improve this. And also we would like to encourage the young, the PhD students, the postdocs involved in this research to connect and collaborate. And we hope that this also, that ELLIS will form a platform for this type of collaborations as well.
Yep, great. Okay, I mean, what I see, there's a lot of positive feedback in the chat window here, and I think there's a lot of positive energy, and I think we should really consider doing this again. So question is how soon? Should it be 1 week? Should it be 2 weeks, and so on? But I think since this is unfolding so quickly, I think we probably shouldn't have a meeting in a month because that would be too late.
I think everything is moving so fast scientifically. We should, in order to keep up with that, weekly or bi-weekly would make sense.
Yep. And I would also like to invite the community to think about forums, especially through ELLIS, to connect to each other. So maybe there are opportunities to research, to validate research, to come up with joint research ideas. So I think we should use this crisis and the fact that we are stuck at home to build these collaborations and build these strong links for the future.
Maybe we could do no, frequent ones, but shorter, because I thought this was fairly long. And so if you do maybe 1 hour or 2 hours in short talks and updates, but more frequently, then I think that would be good.
Yeah, I also think so. I think shorter meetings, more regular, makes sense. So I thought it was good to have this first one a bit longer, I think, to get us all excited and started.
Yeah, different overview. Yeah. I mean, the other area where we also would be interested in feedback is, what is the expertise we need to assemble here? We tried also to reach out to, sort of beyond the canonical community, let's put it that way, and get more other communities from epidemiology and, like Richard Neer's presentation, for example, there's sort of you on board, and maybe there's also, if there are comments about who else, what other views we should get on board. I think that's also important to make sure we solve relevant problems. Yeah.
And finally, I would really like to thank also to Gunnar for bringing us all together and helping us in organizing this. I think many people have been involved, but Gunnar has been instrumental in bringing us together and even hosting us on Zoom. So thank you, Gunnar.
Yeah, I mean, in the end, I have to say, this was Vincent and Daniela who figured it out, but yes, I'm happy to help.
Thanks. Great work.
Okay. Good. With that, I think we can close the meeting for today. So thank you all. Bye.
Thank you, Gunnar. Thank you, Daniela.
Thank you all.
Keep safe.
Goodbye.
Thank you.
Bye.